Okay, thank you. Can you hear me? So I would like to thanks.
Oui, parfaitement.
Okay, I would like to thank President Alba, and I would like to introduce myself. My name is Elena Ambrosini. I'm a researcher working in the National Institute of Health in Italy. And I'm very, very glad to be here. It's an honor for me to be here and share my results of my research with the families, with ELA families. And also I'm the chairman of this session, the second day sessions. And these sessions is focused on vacuolating leukodystrophies that are a group of leukodystrophy that are characterized by the presence of vacuolation into the myelin sheet. And we are going to listen 5 presentation. My presentation that is focused on MLC disease, and then Professor van der Knap with an update on vanishing white matter disease, then Dr. Matthias Eckard of the Bonn University on Canavan disease, and the two last presentation will be on astrocytic Alexander disease. And so I think that we have to start with my first presentation, so not to just okay, so I'm sorry, I hope to, okay. So can you hear can you see my presentation? Okay. Not yet? Sorry. And try to. Oh, I have some problem with my screen. Okay.
Oh, it's coming.
Okay. Sorry, it's my fault. And okay, I'm ready to start. So as I told you before, I am a neurobiologist, so I'm not a clinician. So I'm a neurobiologist working in a National Health Institute in Italy. And my presentation will be focused on the molecular defect in MLC disease. And I would like to give you a message and explain how basic research can help finding therapeutic strategies for this disease. Just to summarize the clinical features of this disease, this MLC, that is the acronym for megalencephalic leukoencephalopathy with subcortical cyst, is a leukodystrophy characterized by macrocephaly, the deterioration of motor function, ataxia, and spasticity. Most patients experience epileptic seizures. Mental decline occurs, but is slower than motor deterioration. And also, some patients experience worsening of clinical condition after fever or minor head trauma. Magnetic resonance imaging is diagnostic. And it's characterized by the presence of cyst in the frontoparietal region of the brain, and also by an increased swelling of the white matter, subcortical white matter, that is indicative of some problems with water exchange in the brain tissue. And the analysis of the pathological tissue highlights that in the outer layer of the myelin sheet, in this cartoon, you can see the oligodendrocyte cell enwrapping the axons. And in MLC disease, in the outer layer of the myelin sheet, there are some vacuolation that are indicated here with that are represented by this balloon, white balloon. So just to remind you, what is a genetic disease? MLC is a genetic disease as all the other leukodystrophy. And the genetic disease means that there are some mutation in the sequence of the DNA that produce a protein. In the normal gene, there is the production, the DNA contains information for the production of a normal protein, but when the gene is mutated, the protein is abnormal or is not produced at all. In the case of MLC disease, there are 2 genes so far that have been linked to the disease. These are the MLC1 gene encoding the MLC1 protein. Was mutations have been found in 70% of patients, and also glial CAM, which is another protein, was mutation have been found in 15–20% of patients. And why this disease these two proteins, the mutations in these two proteins lead to the same disease, because these proteins form a complex that works together. And so that is the reason why this mutation in both of them can lead to the same disease. So just to skip directly to the potential therapeutic option for MLC disease. As you know, this is an incurable disease, but there could be some potential therapeutic option. The first one is, as for the other genetic disease, the possibility to develop a gene therapy strategy. Gene therapy is designed to introduce a normal copy of the mutated gene to restore the function of a protein. And in the last year, there has been an attempt to actually has been developed a preclinical gene therapy strategy by the group of Dr. Estevez and Professor Bosch in Barcelona. And they produce an adenoviral vector able to reintroduce the protein, the MLC1 protein, but also the glial CAM protein, into the brain of transgenic mice. These mice are a model of the disease because they are engineered to not express MLC1, so they are null mice, and they develop some vacuolation in the myelin. By the use of this adenoviral vector injected intracerebellar and intracerebroventricular, they succeed in rescuing the vacuolation phenotype. So this is one strategy, but there is the possibility also of a pharmacological therapy. I put here the question mark, because to go directly to a pharmacological therapy, it's important that we need to know the cellular and molecular mechanism of the disease, because if you have to think that knowing what is wrong in the brain of these patients, you can understand which are the molecular defects, and also you can develop, select and test pharmacological compounds to rescue these dysfunctional pathways. So the story starts several years ago, and I think 15 years ago, when for the first time it was clear that MLC disease was due to a problem in a particular type of cells that are astrocytes. As you can see here, astrocyte is a picture is showing you the population of cells present in brain tissue. The green cells are astrocytes. Then you can see neuron and oligodendrocyte and white cells producing myelin, and also microglial cells. But astrocytes are the cells in which MLC1, the protein whose mutation cause MLC disease, is highly expressed. These are the staining using a specific antibody recognizing the MLC1 protein that show you that MLC1 is highly expressed in astrocyte, and particularly in astrocyte contacting the blood vessels. In the green signals represent the blood vessels, and in the red you can see the MLC1 expressed in this projection of astrocytes that are star-shaped cells embracing blood vessels, and MLC1 is particularly highly expressed. So what is clear that MLC disease is not caused by problem in oligodendrocyte, in the primary problem of oligodendrocyte, but is caused primarily by astrocyte dysfunction. Just to introduce you this type of cells, in this cartoon I've drawn the astrocyte as green cells. These are very multifunctional cells that are responsible for the maintenance of tissue homeostasis. Homeostasis is a very difficult word, but means that they are involved in maintaining the normal condition, the optimal condition, to allow all these cells of the brain to perfectly work. So astrocytes are involved in the control of ion, water, nutrient exchange, also neurotransmitter exchange between the population of cells in the brain and also the external environment. So by doing this, they participate, they take part to, they allow the perfect neuronal function, oligodendrocyte, that are the cells producing myelin, and myelin development. And also they respond to injuries. So the researchers are now trying to answer to these questions. Two questions are important. What's the role of MLC1 in astrocytes? And the second question is, what are the consequences of MLC1 mutation on astrocyte function? These are the two questions that are very important to proceed to the knowledge of MLC disease mechanism and development of possible therapies. So during the last 10 years, a lot of progresses have been made in this field by using a very important model of the disease. The first one is the transgenic mice. Transgenic mice are animals in which researchers have depleted, have avoided the expression of MLC1, so they are MLC1 or glial knockout or null mouse, and the brain and cells derived from these brains demonstrate that there is an increase in the volume of the brain of knockout mice. There are vacuolation, and importantly, astrocytes seems to have a larger dimension. So the volume of astrocyte derived from these mice are larger, bigger than astrocyte. The red line indicates the volume of cells derived from these mice. Another model that have been used in the last year has been the cellular model made of tumoral astrocytoma cells that are easy to obtain and to transform, that have been transformed to express MLC1 wild type. As you can see in this picture, the MLC1 is the red signal that surround all the cells, but when you express in the same cells all the mutants, you can find that the protein is not more able to reach the membrane of the cells, and so it's retained and degraded in the endoplasmic reticulum, that is an area of these cells where the protein can be degraded. And the same cells were used to study a lot of interactors, protein that works together with MLC1 and are involved in the exchange of water and ion between astrocytes and the blood circulation. So another model that gave us some interesting data is the pathological tissue. Coming from other pathologies, not from MLC patient. Why? Because using tissue from different kind of pathologies in this picture, I can show you that the panel A represents the staining, the expression of MLC1 in a normal brain, human brain, that is confined around the blood vessels, as you can see by the red circles. But when you look at the brain where there is a strong inflammation, this is a tissue coming from a patient affected by multiple sclerosis, you can say that there are a huge increase of expression of this protein. What all these experimental models and these results gave us as information? Just to summarize, we can say that all these experiments told us that MLC1 is involved in the change of volume of astrocyte, but astrocytes are a population of cells that undergo dynamic volume changes during their activity, not only in response to injury, but also in response to, for example, intense neuronal activity. So they are cells that dynamically regulate their volume. And it seems that MLC1 is involved in favoring the rescuing of the normal volume of these cells. So when MLC1 is not working or is mutated, we can hypothesize that reactive astrocytes are no more able to perform their normal function. They are not able to support oligodendrocyte and myelin formation, and they can produce toxic substances. So reactive astrocytes, so astrocytes that are unable to gain their normal volume, can also be responsible, for example, for an excessive astrocyte activation and for the worsening of the clinical condition that some patients experience during their life. So the focus of my work and the work of my research group was to study this process, volume regulation and activation of astrocytes, but in human astrocytes. To do that, it's not easy. As you can imagine, pathological tissue is very, very rare. and it's not possible to think of studying astrocyte physiology, pathophysiology using this tissue. So we decided to develop a new approach. This approach is a very powerful technology that has been developed 10 years ago, and the creator of this technology won the Nobel Prize. So the technology is based on the reprogramming of fibroblasts from patients or from healthy control into inducible pluripotent stem cells. These cells retain the ability to differentiate in all the cell type of the body with specific protocols, specific growth factors or activators, and also into astrocytes. And this is what we have done in the last 5 years. We have decided in collaboration with clinicians to recruit skin fibroblasts from patients and healthy donor and reprogram these cells from the skin into iPSC and then into astrocyte with the final aim to study disease mechanism, identify possible target for drug testing and the rescuing of the pathological phenotype. And this is just to show you that we've started with 4 different patients carrying this mutation in the MLC1 gene. Now we have 8 line, and then we have reprogrammed iPSC and tested for pluripotency, and at the end we have obtained astrocytes. This staining, green and red, indicates that these are astrocytes because the staining is performed using marker of the specificity of the astrocyte population. So what we have done with the cells that we have obtained? First, we have tried to find out difference between these cells that are control cells, control astrocytes derived from fibroblasts of healthy donor, and cells derived from patients. As you can see here, there are some clear difference between this population. In patient cells, you can see that astrocytes are larger, so they are larger, the dimensions are different. And they have a lot of vacuolation inside the cell, inside the cytoplasm of the cells, that some of them are indicated with arrows, but there are a lot of these vacuoles in these cells. And this happen in almost all the cellular cells from patients, but we also have 2 out of 6 cellular line in which we observe a higher level of proliferation. You can see there are a larger, more cells in this panel than in the other one. So we can say that astrocytes from MLC patients show increased cell volume, cytoplasmic formation, and increased proliferation capacity. Then we measured, we want to know if the dimension, the increase in dimension of these cells were due to their incapacity to regain their normal volume when they are stimulated with a swelling stimulus. And what we have obtained is that in the control cells, when you apply something that can increase the volume of the cells, like, for example, hyposmotic solution, the patient cells after the first swelling then are able to regain their normal volume. Instead, when you analyze patient cells, you can find that these cells are still swelled and hypertrophic. And just to show you in a more clear way, we applied a stimulus that can induce a swelling of astrocyte, and in the case of wild-type cells, we can obtain a normal volume after 50 minutes, but in the case of mutant cells stay swelled. So we also measured the proliferation of the 2 patients lines that showed a large number of cells, and we obtained that these cells actually are highly proliferating, have an highly proliferating capacity. So we decided to move into the molecular aspect of this phenomenon, of this biological process. As you can imagine, inside the cells, there are a lot of molecules that interact each other to produce the effect of cellular proliferation and cell swelling. But we decided to focus our attention on these molecules because we had some preliminary data on other cell type, but also because this pathway, this cascade of cellular proteins that are proteins with activity is involved in producing astrocyte proliferation, activation, and astrocyte swelling. So we started to analyze the expression of this protein in patient cells. This line, this black line, indicates the expression of proteins that these proteins are belonging to that pathways, but as you can see here in the patient cell line, there are a lot of expression of this protein that is downregulated when MLC1 is functionally is active, it works. So is a huge amount in one of these cell line is quite impressive. As you can see here, this is the control cell line. These are two patient cell line. What does these results want to tell us? They want to tell us that MLC1 in astrocyte is involved in downregulating this cascade. And this cascade, as a consequence of this downregulation, astrocyte can reduce activation as well. But if MLC1 doesn't work because there are mutations, genetic mutation, this pathway is highly activated and it leads to the abnormal activation as swelling and proliferation. So just to show you that these basic research results that allow us to generate human cellular models that reproduce the pathological features of MLC can be a powerful tool to increase our knowledge of the molecular mechanism of the disease and also identify possible molecular targets. So we can we can do a first step towards translational research. Why? Because we can use modulators of these molecules. And behind these question marks, there are a lot of compounds, drugs that are actually in use for the reduction of these molecules that are also implied in other diseases like inflammatory disease or tumoral disease. So our interest is to try to use these drugs, this compound, to modulate the expression of this pathway and try to reduce the pathological phenotype, the cellular pathological phenotype that we have described, vacuolation, increased dimension of these cells, and proliferation. So we hope to do that in the next year because we have this experimental plan in the frame of a project that have been funded by the Italian Ministry of Health. And the main point is to generate a large number of astrocytes from different, carrying different mutation and analyze by using a omics analysis. That is a big analysis that allows us to find different protein expression, but analyzing 100 of proteins and comparing control cells with patient-derived cells, then starting with bioinformatic studies to select and test possible therapeutic compounds that can be screened by functional assay. So just last two slides to make clear that this is a collaborative work, including clinicians, and we have collaborated with clinicians from the Bambino Gesù Hospital in Rome, Professor Bertini Groups, Francesco Nicita, but also Professor Tonduti in Milan, and all his group, and families, and researchers in molecular biology and cellular biology, but also bioinformatics studies are needed to select possible modulators of the molecules that we found downregulated or upregulated, with the final goal of the identification of the most appropriate strategy to arrest or slow down disease progression. So just to start with my initial question, how basic research can help to find a cure. Basic research helps to understand the cause of the disease and how nature works. Then this information can be used to develop preclinical therapeutic strategies that can then applied to test and tested in clinical research. So I would like to thank you for all your attention, and this is the, the panel showing all the collaborators, and I would like to thank all my group in Rome. So I finished my presentation, so I think that if I'm not wrong, question can be grouped at the end of the presentation of the other speaker. Please, Elise, confirm me that we can go on with the presentation. And I think yes, that there is a question. There is a question time at the end of the presentation. So it's my pleasure to present Professor Marjo van der Knap to introduce you. I think that she doesn't need any introduction because she is one of the most renowned scientists in the field of leukodystrophy. And her talk will be focused on vanishing white matter disease. So please, Marjo.
Thank you so much for the kind introduction, Elena. I'll try to share my screen. Yeah, you all see my screen?
Yes, we can see your screen.
Okay. Good afternoon. It's a pleasure for me to participate in this ELA meeting for patients and families. And the subject of my talk is vanishing white matter, also called CASH. So this is my research funding. It causes no conflict of interest. The VU University Medical Center holds a patent on guanabens treatment in vanishing white matter, and we also have an orphan designation. I'm a consultant on the subject of the vanishing white matter trial development for Calico, and I'm a co-investigator for the Alexander disease trial. This is the contents of my talk. I'll give a brief overview on vanishing white matter, disease mechanisms and therapy targets. I'll introduce the International Clinical Consortium. I'll give an update on the guanabens trial. I'll introduce the EIF2B activator trial. And I'll have some closing words on the International Vanishing White Matter Registry. So vanishing white matter is a highly heterogeneous disease and it can have its onset at all ages. Classic vanishing white matter has mostly its onset between 2 and 6 years and mostly leads to a chronic motor decline like cerebellar ataxia and also spasticity. Cognitive decline and epilepsy are less obvious. In addition, there are episodes of severe decline of neurological function, also with irritability and lowered consciousness. And these episodes of rapid decline are provoked by physical stresses like febrile infections and minor head trauma. They are followed by partial or complete recovery, or patients may die during an episode. And death typically occurs after a variable number of years to decades. Early onset vanishing white matter has its onset before the end of the first year of life and is a very serious disease. It's characterized by a general encephalopathy with no or little development. If patients have development, there's a fast regression. There's severe epilepsy typically and irritability. And until now, all patients have died before the age of 2 years. Late onset vanishing white matter has its onset in adulthood and is different and is especially characterized by cognitive decline and behavioral changes, and later and less striking motor decline. The clinical course is typically slow, and stress-provoked episodes of rapid decline are few. And these patients mostly live for decades. So the disease onset is the best predictor of the disease course. It defines the type of disease, and it also and the rate of progression. Oh, what did we did a natural history study that was published, I think, in 2018 on almost 300 patients. And because we knew the effect of the age of onset, we divided the patients into 6 groups. onset below the age of 1, between 1 and 2, 2 and 4, 4 and 8, 8 and 18, and from the age of 18 on. And if you look at these Kaplan-Meier curves, and on the x-axis is the age, then you can see that with the later onset, the walking is longer preserved, cognitive decline is later, and survival is longer. If you, however, look at disease duration, then you can see that the very early onset disease has a rapid disease course, and that becomes slower with later onset. That's also true for survival, but for cognition, you can see that adults in green have a relatively bad course. This looks like all very reliable, but actually vanishing white matter is extremely heterogeneous. So in yellow, for instance, you can see that some patients die very soon and some patients still live in their 40s. So it's an extremely variable disease. Sex doesn't matter, males and females is approximately the same, but having episodes of rapid decline matters. So without episodes, survival and loss of ambulation is slower. The survival is longer and loss of ambulation is later. And with episodes, it's much faster, and that's independent on the age of onset. Vanishing white matter is caused by a genetic defect in an enzyme that's called EIF2B. Now, EIF2B is here, and that's conditional for the translation of DNA into new proteins. So it's conditional for protein synthesis per se. It also regulates the rate of protein synthesis, and with that it's important for the cell stress response, because during stresses, protein synthesis needs to be downregulated, and I'm going to show you that. In vanishing white matter, mutations decrease the activity of this enzyme, but never to zero, because since EIF2B is conditional for protein synthesis, and you cannot live without EIF2B activity. This is the integrated stress response. That's here. And what you can see is stresses, infection, reactive oxygen species, fever, amino acid starvation, and misfolded proteins. They all activate different enzymes. These stresses activate different enzymes, but the enzymes do the same thing. They phosphorylate EIF2. And when EIF2 is phosphorylated, it tightly binds EIF2B, which is our factor. That has two downstream effects. One is general protein synthesis is downregulated, that's what I told you. And the ATF4 transcription response is activated. And this transcription response contains many different factors. And these factors are going to help the cell repair and proliferate again. And if that fails, they help the cell to go into apoptosis. There's a feedback loop that makes that the whole situation goes back to normal. And GAT34 is the entry of the feedback loop. Now, in vanishing white matter, EIF2B activity is always down. The activity is clearly sufficient to have a normal general protein synthesis. We never found a defect in general protein synthesis. But the ATF4 transcription response is constantly activated, meaning that cells in the body think that there is stress. And whereas there is apparently no stress. And we think this is the pivotal pathomechanism of vanishing white matter. That means that we now all of a sudden have a number of therapeutic targets. And that's what Elena also told you. If you know the pathomechanism of the disease, then you can look at which targets you can modulate. So obviously, the most important target would be EIF2B itself. Can we activate it? Another target would be the GAT34 feedback loop. Can we also activate it or modulate it? And then, of course, you can think of the ATF4 transcription response, because there are many different factors here that could be modulated. And that means that we all of a sudden have potentially multiple drugs for each option. So a lot of things to work on. And when I started to realize that, because I got approached by many different companies, I realized the problem. because the disease is too rare to have multiple parallel trials because they will compete with each other and each will be underpowered. So that's why we founded a consortium of clinical experts on vanishing white matter, because we want to facilitate therapy development and guide trials. Because we would like to test multiple drugs in overlapping or parallel trials, and that these trials are adequately powered. And an easy way, and nothing is easy in this field, but the most obvious thing to do would be to share placebo controls, because such trials are typically placebo controlled. So part of the patients get the real drug and part of the patients don't. And we don't know which patient has the real drug and which patient doesn't. And we analyze the differences at the end. But if these trials would share the placebo-controlled group, then that would reduce the two advantages. One is reduce the number of patients needed for the trials, and also minimize the number of patients on placebo. And we thought a good means for that would be to develop a core protocol that could be shared by all trials. This is the Vanishing White Matter Consortium. You can see that it is based in Europe and North America, Canada and the USA, and contains Joshua Bukowski from Salt Lake City, Adeline van der Ver from Philadelphia, Brent Vogel from Los Angeles, Ingeborg Kregelo-Mann from Tübingen, Enrico Bertini from Rome, Geneviève Bernard from Montreal, Ali Fatemi from Baltimore, and Nicole and I from Amsterdam. And our goal is facilitate therapy development and guide trials. Then I'll talk about the update of Quanabens. Quanabens is an alpha-2 adrenergic antihypertensive drug. It entered the market in the 80s, so it's long known, it's safe, it's FDA approved, and it's no longer used because they are better antihypertensive drugs. It also impacts the ISR through the GAP34 feedback loop. And what we found we have mice that have vanishing white matter. They have the mutations, they have the brain pathology, they have the clinical picture, this motor disease. And in these mice, we tried Quanabens and found it ameliorates ATF4 activation in a dose-response dependent manner. So what you see is the higher you dose Quanabens, the lower the ATF4 transcription response becomes. So that's good. It ameliorates neuropathology, and then it also improves the motor performance. So these mice also get ataxia. This is the normal mouse. This is the vanishing white matter mouse, and you can see that there's a lot of slips on the balance beam. So its motor performance is not that good. This is weekly Quanabens, this is daily Quanabens. You can see that the motor performance becomes much better, and with daily Quanabens, it's just normal. Then time on the balance beam. How long does it take a mouse to reach the other side? This is the balance beam in normal mouse, and you can see that the vanishing white matter mouse takes much longer, and that with weekly Quanabens it becomes better, and that with daily Quanabens it's normal. So that led us to a Quanabens trial, and that's relatively easy since Quanabens was already in use. Sorry for my clock. And so we decided to start a trial, and it's a single center trial, but it's international. We started in May 21. Inclusion will end in 24, and the end of the trial will be in 25. It's open label with historical controls, so no placebos. We can have 40 vanishing white matter patients, and we decided to go for the early onset form. So onset below 6. Patients need to be able to still walk 10 steps without or with light support of one hand and have a maximum disease duration of 8 years. Or we give Quanabens in the evening to avoid side effects during the day, and it's known for its rapid tolerance. So we also saw that in mice that the first side effects that we see on day 1 are not seen on day 2 anymore. So that's why we start patients on the low dose, and we titrate them in a couple of weeks to the maximum tolerated dose. Until now, we have 26 patients included. I think there are 2 more right now. We have 2 dropouts, one because of a disbalance between the intensity of the trial and the mild neurological deficit, and one because of personal circumstances. and they are all young patients. Guanabans is well tolerated. Children resume their normal daily routines. They have subjective positive effects on general well-being, have more energy, they can walk longer distances. The most important side effects are alpha-2 adrenergic and so expected, which is sleepiness, dry mouth, feeling unsteady, tremor, constipation, nausea and vomiting. We also had some unexpected side effects like sad feelings, vivid dreams, nightmares, and sometimes even hallucinations, and some patients become hyperactive. But these side effects are transient and they resolve in a couple of weeks or months. Some last longer and some come back. And sometimes we need extra medication like laxative, antiemetics, risperidone, propranolol for the tremor. And sometimes it helps if we split the Guanabans dose into smaller gifts. And of course, we need to have a longer follow-up and complete the trial to evaluate the efficacy of Guanabans as measured by clinical outcome, quantitative MRI parameters and biomarkers. But until now, it's safe. Then I have the pleasure to announce another trial, and that's an EIF2B activator trial. And the drug is called ABBV CLS 7262, so I won't repeat that again. And it's developed in a collaboration between Calico and AbbVie, and it's a potent small molecule that activates EIF2B. It can be measured in an enzyme assay, and as you can see, these are normal cells and these are mutant cells from patients. And this is the enzyme speed, and it becomes higher in treated normal cells, so the conversion rate becomes higher. And in patient cells, you can see that activity is slow, and with treatment, it mostly becomes normal. Then this drug was also tested in vanishing white matter mice. And what you can see here is in the time to cross the balance beam, just the same test as we saw before. And you can see that it becomes longer in vanishing white matter mice with increasing age, and that with the drug, it becomes in the range of normal. So that also looks very good. Now Calico, in collaboration with AbbVie, is developing this drug as a potential treatment for vanishing white matter and is partnering with the Vanishing White Matter Consortium on this subject. Calico will use the core protocol for their trial development, so that's really beautiful. They have tested the drug in more than 100 healthy people, treated 14 days max, and they administer it by mouth. And the events are non-serious and similar as in ALS, which I will discuss below. It increases EIF2B activity, inhibits the ISR and blood cells from these healthy volunteers, and it enters the cerebral spinal fluid in a concentration you would like to have it. The first trial with this drug was in patients with ALS, and 31 patients are included now, and the longest treatment period is now more than 1 year, and it's a placebo-controlled trial. It's still blinded, so the preliminary safety information is on blinded patients, so we don't know whether the side effects are related to the placebo or to the drug. 10% of the patients report headache, 7 nausea, 7 itchiness, 7% constipation, and 7% dizziness. So this is a very good safety profile. A small phase 1b/2a clinical trial evaluating the drug in vanishing white matter has been initiated. So it's an open label phase 1a to phase 1b/2a trial, and it's the first study in vanishing white matter, and it's part of a larger vanishing white matter clinical development program. The study will evaluate safety, tolerability, pharmacokinetics, and exploratory efficacy. And the study has already started in the USA, where it enrolls adults. And the study will be amended to open in Europe and include adults, adolescents, and children by the end of this year. And the Vanishing White Matter Consortium aims at the inclusion of more severely affected patients that would not qualify for the phase 2-3 clinical trial. That's also going to be there. So the results from the phase 1b/2a study will be used to inform the future phase 2/3 clinical trial. And if you want to be updated, then please look at the clinicaltrials.gov website, where which will be modified as needed and as frequently as needed. And so you can find there the most recent information. Then my final subject is the International Vanishing White Matter Registry that we started in 2004. It now contains 420 patients. And what's important to realize is that although the disease mostly has its onset below the age of 6, as you can see here, more than 6 to 12, 12 to 18, and above the age of 18, that the number of living patients is mostly 18 years or higher. And that's because the younger patients die much sooner. So most living patients are above the age of 18. We are working on maximizing the impact of the Vanishing White Matter Registry, because this registry is crucial for several studies. The natural history study, which is still running. We are executing a study on the impact of the disease on healthy family members. We are collecting historical control data for trials, because especially in the younger children, you don't want placebo-controlled trials, because the patients can deteriorate so rapidly. We are searching. The registry allows us to search for eligible trial candidates easily. We can use the registry to search for valid trial endpoints, and also we can use the registry for post-marketing studies. We are in progress to adapt the governance. It used to be an Amsterdam-driven registry, but we want a central role for the Vanishing White Matter Consortium and patient advocates. We want to adapt the registry to facilitate data reuse and data sharing with other researchers and with industries. So we are working on a new IRB approval and reconsenting patients. We are working on a secure portal for data sharing and data release for analysis. And we are working on regulatory readiness. So we already have received a fee waiver from the EMA qualification procedure, and we have applied an EMA innovation task force. And the input of patients and families is, of course, extremely important. The registry is more valuable with higher patient participation and higher numbers. So I would like to show you the Vanishing White Matter team in Amsterdam. This is the trial team. We have molecular biologists, neuropathologists, biomarker persons, pharmacists, MR persons, and then also people that are working on with me on the trial and the registry. I would like to thank you very much for your attention, and I would like to thank the ELA for their support and also for letting me talk. Thank you.
Thank you, Marjo. I'm very impressed by this presentation that gives hope to patients affected by this disease, and also highlight the importance of the generational consortium and the registry and the collaboration of different expertise, starting from the molecular biology to the clinicians to pharmacologists, and so I want to encourage people from the audience to send also question on the chat, because we can answer during the presentation. And for all the other questions, we wait the question time. So it's a pleasure for me to present the next speaker, that is Matthias Eckert from Germany, of the Bonn University. And the title of his presentation is "Does neuropeptide N-acetyl-aspartyl-glutamate acid plays a role in the pathogenesis of Canavan disease?" So Matthias, it's up to you.
Yeah, thank you. I'll share the screen. So, okay, thank you very much for the introduction and for the opportunity to present my research, or our research, on the project that is currently funded by ELA. So it's an ongoing project, and has not come to an end. So there are some preliminary data I will present. And so my topic is Canavan disease. I'm not a medical doctor, so I'm doing basic research. So therefore, I will not talk too much about clinics and symptoms in human patients, but I will focus on the mouse model on the Canavan disease. Okay, so Canavan disease is leukodystrophy, also characterized by spongiform degeneration, as in MSC1, for example, as presented already. And the genetic disease, it's a autosomal recessive disease, and the cause of the disease is a deficiency in an enzyme called aspartate or aminoacylase 2. And this enzyme is encoded by the gene which is called ASPA for the aspartate gene, so called in short ASPA. And deficiency in this gene and in the enzyme activity leads to the accumulation of the substrate of this enzyme. That's the N-acetyl aspartic acid. That's this compound here. So that means the ASPA enzyme is deficient, and then it comes to an accumulation of this compound. So there were some disputes in former times. What is the cause or the reason now for the disease, for the leukodystrophy? And some research by our group, but also by other groups, showed that it's really the accumulation of this compound, this NAA and acetyl aspartic acid, that is toxic for the brain. An alternative explanation was that the degradation of the NAA leads to the release of acetyl groups in oligodendrocytes and myelin-forming cells, and that these acetyl groups are important for forming myelin. But there are animal models that showed that this is not the case in the disease, at least in the animal model, I have to say, because we did not study human patients. So the idea was that the NAA is degraded by the ASPA enzyme in oligodendrocytes, and this acetic acid is needed for myelin formation. But if we look at a mouse model which is not able to produce any NAA, that's a mouse model that is deficient in the NAA synthase or NAT8L gene, then this mouse look almost normal. And if we have a mouse that is deficient in the NAA synthase and also is deficient in the aspartate, so it means it has a Canavan genetic model. If you have a Canavan mouse that is also deficient in the NAA synthase, we don't see the spongiform degeneration in this mouse. So this is here on the left lower part. This is a Canavan mouse model, where you can see the spongiform degeneration. That's a normal wild-type model on the top. And this is a mouse model which has the deficiency in the ASPA and also a deficiency in the NAA synthesis. That means it's really the accumulation and not the absence of degradation product of the NAA degrading pathway of the ASPA that is responsible for the disease. So that's earlier, these are earlier results that we have found. And now if we look at the therapeutic options that are possible for Canavan disease, one, of course, would be to deliver the active ASPA gene or the aspartate, the active enzyme, as in the other diseases that are already mentioned. And the other possibility, potential possibility for therapy would be to inhibit the synthesis of NAA. That would be a substrate reduction therapy that is already known, especially in the field of lysosomal storage disorders for some diseases. There are some research groups trying to perform this delivery of active ASPA, and there are also research groups who are trying to find inhibitors for the NAA synthase, for the enzyme that produces the NAA. And so some trials and some inhibitors have already been identified, but to my knowledge, there's no really candidate inhibitors that can be really used already in clinical trials. Okay, the topic of the next part of or the second part of my talk, and the topic of the project is currently funded by ELA, is the following. That results from the fact that not only NAA can accumulate in Canavan disease, but there are also reports on the accumulation of a product that is formed from NAA, that is a peptide that is called N-acetyl glutamate, NAAG in brief. And this NAAG or NAG, we can call it, it's a neuropeptide produced by neurons and is formed from the NAA by adding glutamate residue to this compound. And this is a reaction that also occurs like the synthesis of NAA in neurons by enzyme called NAG synthetase. And these NAG synthetase enzymes are encoded by two different genes called RIMK-LA and RIMK-LB. Why they are called so, it's not so important for us. There's some historical reasons. And now the question in our project was, does this peptide, NAAG peptide, the NAG peptide, somehow contributes to the pathogenesis of Canavan disease in the mouse model? The reason why we have asked for this is that the NAG peptide is also a source for NAA, because NAA is a precursor of the NAG. So NAA is, glutamate is added to the NAA neurons, then NAG is produced, and the NAG can be released from neurons. And then an enzyme that is produced by astrocytes that also have already been introduced in the previous talks, an enzyme produced by these astrocytes can then cleave off the glutamate residue again, and then we have N-acetyl aspartate again. That means via the NAG peptide, we can also theoretically also increase or get to an increase of the NAA concentration in the extracellular space of the brain. And our question was, does this release of NAG and the degradation of NAG somehow has contributes to the pathology of the disease and somehow maybe contributes to the increase the accumulation of the NAA in Canavan disease in the mouse model of Canavan disease. Towards this aim, we have generated mice that are deficient in the NAG synthase genes. There are two genes, as I mentioned, RIMK-LA and RIMK-AB. The RIMK-AA gene seems to be the more important because we could show that if we knock out this gene, then we lose most of the NAG in the brain in the adult animals, whereas the RIMK-AB gene seems to be more important for early developmental stages of the brain development and also for some other reactions. It means the RIMK-AB may be more important than the RIMK-A. RIMK-A may be more important than the RIMK-AB in terms of the NAG synthesis in the adult brain. We now cross these mice that we have generated with the ASPA knockout mouse, which is aspartate knockout mouse, where we use mutant mouse called NUR7. That's a natural mouse mutant that have been identified, which also develop the Canavan disease symptoms. And if we knock out RIMK-AA in this mouse line or RIMK-AA and also the RIMK-AB gene, we get double and triple deficient mouse models. And the first thing what we have looked at was the survival. So the survival rate was not, let's say, was not decreased in the double and triple knockouts, but it was even, the survival rate was even a little bit better than the conventional aspartate knockout mouse, which could be a first sign that maybe there could be some improvement in the pathology in these double and triple knockout mouse. But if you look at the body weights of the mice, we see no clear difference. We have reduced body weights in the Canavan disease mouse model, and also in the double and triple knockouts, we have the same reduction in the body weight. So there was no strong difference. Then we did several behavioral studies with these mice. We performed a so-called open field test. where simply the mouse is monitored how it moves in an empty area for about 20 minutes. And we measured the total activity, the distance the mouse migrates during the session time, and the average velocity. And there was no difference between the different Canavan disease mouse models in this parameters. Only one parameter, that's so-called rearing activity, how often a mouse stands up to the hind legs. And there's a very sensitive parameter for the Canavan disease mouse model that this time is much reduced in the Canavan mouse. And if we look at the mouse which has a Canavan mutation and has also mutation in both NARC synthetase genes, then there seems to be a tendency towards higher activity, rearing activity. It's of course because unfortunately we don't have so many animals currently. We need some more of these. It's an ongoing project, and therefore this difference between these triple knockouts and the conventional ASPA knockout mouse is not statistically significant, but we see some tendencies that there's some slight improvement. Then we looked by some other behavioral tests for the muscle power, the grip strength. It's measured with this device here and the mouse, and then you simply check which force you can apply or the mouse can apply and to this device here. And there we see almost a normalization of the grip strength in the triple deficient mouse model. Whereas, in contrast to this, we looked also how the mouse moves, how it behaves, and we calculated a so-called ataxia score. And there we don't see a big difference between the different Canavan mouse models. Also in the so-called grid test, where the mouse is placed on a grid and then the grid is turned upside down, and the time is measured, how long the mouse can hold on this grid. With the cut-off time of 1 minute, there we don't see really a difference between the different Canavan disease mouse models. All the Canavan mouse models perform worse than control mice, but between the different Canavan disease mouse models, there's no significant difference. Okay, also the spongiform degeneration. This is very preliminary data that we show here. We have only one or two mouse brains analyzed per genotype, and this is an ongoing project, but we still see the spongiform degeneration in the double and also the triple knockout mice. So it's difficult to say currently whether there is any clear difference in the degree of spongiform degeneration. Okay, this we skip. And another parameter is the activation of astrocytes, already mentioned in previous talks, and the GFAP was also mentioned. That's a marker for astrocyte activation, and we performed also immunofluorescence staining as presented in previous talk, but also Western blot analysis, where we simply looked at the total amount of this GFAP protein by a biochemical method. And there we found that in cerebellum and stem brain, there's no difference between the different Canavan mouse models. But in the forebrain, if we take the whole forebrain of these mice, then there seems to be a slight reduction in the amount of GFAP in the brain of this triple deficient mice. Okay, and then we, at the last part, we then measured, of course, the concentration of the NAA, the N-acetyl aspartic acid. For this, we developed a new enzymatic assay. Currently, in most studies, the NAA concentration is measured by magnetic resonance spectroscopy or by mass spectrometry. And we thought, because sometimes it's, for basic research at least, it's sometimes important to have a very rapid assay to perform this determination of NAA in a few hours without waiting for the time slot for the mass spectrometry, for example, or MRS. And therefore, we simply expressed the ASPA enzyme in bacteria and used now the ASPA enzyme the active ASPA enzyme also for the determination for the of the NAA concentration in brain tissues from our mouse models. I will not go into many details here. On the right part, for the people who are some are familiar with chemistry, can maybe imagine how this assay works. To make it briefly, if we simply take the NAA, we incubate the sample, we take the sample with the NAA, we simply incubate the sample with the ASPA enzyme, then aspartic acid is released, and there are already established enzymatic assays for determination of the aspartic acid. And that's simply then done in a very easy photometric way within this assay. If we measure the NAA concentration in our mouse models in the forebrain and the cerebellum plus stem brain, we see in the forebrain no significant difference, no reduction in the NAA concentration in our double and triple knockout mice. And in the cerebellum plus stem brain, we see, let's say, a slight tendency towards lower NAA concentration in the triple knockout, but this is currently not statistically significant. So we can currently not say whether there is a real effect on the NAA concentration in the triple knockout mouse. Okay, what I briefly would like to mention is one fact. I've mentioned in the beginning that the absence of NAA synthesis in the Canavan mouse model leads to the absence of the spongiform degeneration in the brain, but unfortunately it does not in the mouse model leads to a significant improvement of survival time. That's because the NAA synthase deficient mice show, irrespective of whether they have active or inactive aspartate gene, they show a phenotype of very premature death. So they die suddenly without any signs, clinical signs before, and that leads them to a very premature death, which is not statistically significant from the premature death of the Canavan disease mouse models. And now what we have generated some other mouse models, which are shown here on the right side. That is simply there we followed the survival or the number of animals that are still alive. And we followed here about 8 to 10 liters of different mouse litters that we have generated from the different mouse lines, and simply calculated or simply counted how many mice are alive after 3 weeks at the time of weaning and after 3 months. That's the orange bar. And the mouse model that we have generated here is now not an ASPA knockout mouse, but an NAA synthase knockout mouse line, which has in addition deficiency in the NAC synthase gene RIMK-LA or RIMK-LAB, or in both. And you can see that there seems to be premature death in the normal NAA synthase knockout mouse, which still have RIMK-LA, RIMK-LAB, and also in the NAA synthase knockout mice that lack RIMK-LAB but still have RIMK-LA. There's loss of animals, and also in mice that have only RIMK-LAB but no RIMK-LA, and also no NAT8 and no NAA synthase. That's also premature death. But if we knock out all 3 genes, NAA synthase, RIMK-LA, and RIMK-LAB, there's no premature death of animals. That somehow is somewhat very strange, because one would believe that the NAA synthase makes NAA, and then the RIMK-LA, RIMK-LAB genes are responsible to make the NAC peptide, which is derived from NAA. That means in this case, we don't have any NAA, so why should the knockout of the RIMK-LA and RIMK-LAB gene that use NAA to produce the NAC peptide should have any effect, because the NAA is missing here. The explanation of possible explanation for this is that the NAC synthases also produce another compound that is called citral glutamate. So that means if there's no NAA, but also even if there is NAA, they use also citric acid and add glutamate to the citric acid, and then they produce citroglutamate. If there's no NAA, as in the NAA synthase knockout mice, it seems that the enzyme, the RIMK-A, RIMK-AB encoded enzymes, then use citric acid more because NAA is missing, and they produce more citroglutamate. And that's what we could also measure in the knockout mice. So in the normal wild-type mice, we see a certain concentration of citroglutamate, and in wild-type mice and in the NAA synthase knockout mouse, we see an increase of the citroglutamate concentration in the brain. And our hypothesis currently is that this increase in citroglutamate concentration is responsible for the premature death of the NAA synthase deficient mice that we don't see if we also knock out RIMK-A and RIMK-AB, because then citroglutamate is no longer produced. Okay, to sum up the results of our study, as I mentioned, it's an ongoing study and we don't have yet all the histological analysis finished. And also we want to analyze the degree of the pathology in more detail, but therefore we need some more animals which are currently bred in our animal facilities. But we can already say that we found only very weak or no significant effect on the total NAA concentration in the brain of the Canavan disease mouse models if we delete also the NAC synthases. There's some small improvements of the behavioral level of these mice, but it's only very small and very weak improvements, and therefore inhibition of NAC synthesis alone, at least, is most likely not a therapeutic option of Canavan disease. An important point about was that there's also no worsening of symptoms and behavioral parameters of the mice lacking NAC synthetase. That means that inhibition of NAC synthetase in parallel maybe to NAA synthase inhibition may be potentially reduce side effects, which could be a possible outcome of the use of inhibitors of the NAA synthase, because as I mentioned, as I showed, that the NAA synthase knockout mice have this premature death phenotype, and this of course could be a problem if we use inhibitors for the NAA synthase as a therapeutic option for Canavan disease, and in that case maybe NAC synthase inhibitors could be somewhat relevant. Okay, finally, I would like to thank you for your attention, and I would like to thank the people involved in this work. Mainly Yvonne Becker, Liwa Wang Eckert, and Volkmar Gieselmann have been involved in this work. I would like to thank the ELA for financial support of this project, and I would also like to thank the DFG, Deutsche Forschungsgemeinschaft, who had to support the generation of these initial analysis of the NATED-L, RIMK-A, and RIMK-AB deficient mouse models. Okay, thank you.
Okay, thank you very much, Dr. Heckard, for this very interesting presentation. And I have to say that the last two presentations highlights that the mouse models are still important, and for this leukodystrophy, just not only to study the molecular mechanism, but also for testing therapies and therapeutic compounds. So it's important that the animal model is able to represent or recapitulate all the features of the disease, and so it's not easy to have a good model in some way in this leukodystrophy. So thank you very much again, and I would like to ask the speakers to check the chat, because there are some questions in the chat, and I would like to know if there is the possibility to translate the question that are written in Spanish or in French in English. Maybe our translators could have access to the chat. I don't know. Anyway, it's my pleasure to go on and present Angela Gritti, Dr. Angela Gritti, and Dr. Vasco Meneghini from the Hospital San Raffaele in Milan. And this is one of the main European Center for the Study of Rare Disease and Genetic Therapy. And so Angela Gritti is one of the most renowned scientists in this field. So it's my pleasure to start with their presentation that will deal on the development of editing technologies for the treatment of Alexander disease.
Okay, thank you, Elena. I will share my screen. Okay, I hope that everything is fine.
Yes,
Okay.
we see you.
Okay, thank you. So good afternoon to everybody, and I really have to thank ELA for inviting us to this meeting, and I'm really glad to give you an update on this project on Alexander disease that was funded by ELA. So, and I'm also grateful to the previous speakers because they introduced some basic concepts on astrocytes and GFAP, and so this makes my job a little more easier to describe this presentation. So Alexander disease, just a very brief introduction. Alexander disease is a primary disorder of astrocytes that is caused by gain-of-function mutations in the genes encoding for the glial fibrillary acidic protein. So gain-of-function means that you just need one copy of the mutated genes in order to manifest the disease. So this mutation, the mutated protein, leads to an aggregation of the protein itself and a reactive response in astrocytes with severe effects on the central nervous system. Despite many efforts and also promising results that have been obtained in the past few years, there are still many aspects of this pathology, of this disease, that are still unclear. Particularly, for example, the initiating events in the accumulation of the mutated protein and the astrocyte pathology, but also the relationship, for example, between astrocyte dysfunction and the myelin defects, and also this variability in the age of onset and in the disease severity that is present in the patient. So the most, let's say, straightforward idea in view of a therapeutic approach is that of reducing or eliminating the production of the mutated proteins, or the protein that initiates this disease process. And a possibility is to achieve a generalized suppression of this protein, so reducing both the mutant and the wild type, the normal protein. This is the mechanism of action of antisense oligonucleotides, which interfere with the production of the protein at the level of the messenger RNA. And so this reduce and disrupt, let's say, the production of the protein. So this approach has been tested in mouse model of Alexander disease with good results, and is currently under clinical testing in patients. So what we are proposing is a different approach based on state-of-the-art gene editing technology. These technologies are more or less using the same platform, which is called the CRISPR-Cas9 platform, but with a different, let's say, purpose. So in one case, in this first approach, the CRISPR-Cas9 nucleases, the final outcome is a break. So the aim is to cut, let's say, the DNA, while in the second approach, they are called base editors, the aim is to correct the DNA, so to correct the mutation. So using in the first approach, we have this enzyme which is called Cas9. Here you see it in gray. So this Cas9 protein is able to cut the DNA, and this is coupled to a sort of guiding. There is something. This is coupled to a sort of a guiding system that is called the single guide RNA. So basically, this system gives the specificity to the system because you you can design these guides in order to really match specifically the portion of the DNA that you want to cut. And of course, in our case, we want to cut the gene encoding the mutated GFAP. So the results, the resulting breaks in this DNA strands are not faithfully repaired. So this results in a stop signal that hamper the production of the protein. Okay, and this is the first approach. While in the other approach, so the base editors, we still have the guiding system, recognizing the specific sequence of the DNA that we want to target, but we have a modified Cas9 protein that is unable to cut the DNA, but only in this case delivers another enzyme that drives a very precise change in the DNA sequence in order to correct the mutation. So using these approaches, we should obtain a stable downregulation or a stable correction of the target protein. Both of these approaches are compatible with the idea of a one-shot treatment. And importantly, both these approaches allow us to discriminate between the mutated and the non-mutated GFAP. So instead of suppressing both. The main risk associated to this system is actually that we may target genomic sequences that are not the one that we want to target, but that display high similarity with the sequence that we want to target. And this is called off-target activity, and this may result in genotoxicity. So based on these premises, our longer-term goal is to develop a definitive single dose, so one-shot therapeutic approaches for the treatment of Alexander disease, taking advantage of these gene editing approaches. And in this pilot project that was funded by ELA, our main goals were to set up the reagents and the tools to do this, and to also to provide a proof of concept using in vitro and in vivo models. And the key person in this project is Dr. Vasco Meneghini, a project leader in my lab, and Dr. Meneghini is here today, connected, and of course he will participate to the discussion. I will now show a summary of the main results that we have obtained and the future perspective, of course, of this project. So first of all, we had to generate cell models expressing the wild type or the mutated GFAP gene. And we started using fibroblasts because they are easy cells to be cultured, to be maintained in culture. So we generated the vectors, lentiviral vectors encoding for the GFAP, the normal GFAP protein, or the GFAP protein carrying these mutations, which is one of the most common mutations in Alexander disease. And this GFAP protein was linked to a red fluorescent protein that we can detect using dedicated machines. So if the fibroblasts are not treated with these vectors, they are, let's say, not red, and they cluster here. While when you transduce, when you treat these fibroblasts with these vectors, they start to produce this red protein, and you can see that they cluster here, and so they are bright red, and you can see that more than 90% of these cells become bright red. So this was important to set our sort of detection system. Of course, then we wanted to use these bright red cells in order to see whether to test the efficacy of our system, so the Cas9 enzyme plus the guides RNA, if this system were able to disrupt, let's say, the expression of the GFP. So the idea is that if you disrupt the expression of the GFP, you should have a decrease in the bright red cells, in the percentage of bright red cells. And this was actually the case. So if you start with 90% of red cells, and then you treat these cells with this Cas9 plus the guiding system, this is a transient delivery of this system, you end up with a significant reduction in the percentage of red bright cells. So meaning that this this means that you have really silenced, the GFAP expression. So the next step was to design and select specific guides that could target not only the normal, but also the mutated GFAP genes and discriminate between them. And I report here only one example. So this system, the Cas9 with the single guide RNA, this is disrupting the gene far from the mutation, so it affects both the wild type and the mutant protein. And again, you see that upon transient delivery of this system, you can, in this case, obtain more or less, again, 50% of reduction in the percentage of the cells. And I have no time to show you now, but this screening has been done with a lot of guides. And of course, we have also selected guides that are able to silence specifically the mutated GFAP gene while sparing instead the wild type form. So, so far we have, let's say, demonstrated that we can transiently deliver our system gene editing machinery, let's say in fibroblasts, obtaining this reduction of GFAP expression with this CRISPR-Cas9 nuclease system. So the first approach. So what about the other approach? So the base editors, are they similarly effective in reducing this expression of the GFAP? So the first thing here that we did was again to test different single guide RNAs that could target the GFP mutation. So delivering, in this case, the base editor, okay, to the right DNA sequence that we want to target in order to promote the correction, in this case, not the cut, but the correction of the DNA sequence. And we screened a lot of different guides and we selected some of them. This is again one example of one guide that delivered together with the Cas9 to this specific sequence of the mutated GFAP gene is able to promote the conversions of the sequence with an efficiency of the 35% more or less. So meaning that we can rescue, let's say, the correct sequence with this percentage. So this is a promising result. For sure, there is room for improvement, but we can do this. At least we know that we can do this. So of course, I mentioned before, there are still some safety concerns that have to be addressed. So the potential side effects associated with these technologies, and there are ways to check for this off-target activity. These are quite complex graphs, but the take-home message is that if you focus here, you see that so far with our technology, with our detection, we can say that the majority of the target is on target. So we can really have very specific targeting of our DNA sequence, very low undetectable off-target activity, not only with the first approach with the nucleases, but also with the base editors. Of course, these are data that have to be confirmed, but at least this seems to be promising. So all this work has been done on fibroblasts, but of course fibroblasts are not the target cells. So the target cells are astrocytes in Alexander disease. So the next step was to verify whether this system could really work in astrocytes. So we started with the murine astrocytes, of course, that we derived from murine neural stem cells. Again, we treated these cells with this vector that I showed you before, So we have these bright red astrocytes. And for this experiment, we decided that we should deliver this gene editing machinery, not transiently, but we wanted to deliver them in a stable way. And to do so, we decided to use adeno-associated vectors because these vectors are known to have a nice tropism for neural cells. They are known to give a stable expression of the transgene, and also they are already used in many different experimental settings, and also in clinical trials now in patients for several neurological disorders. So we delivered our Cas9 with this gene editing machinery using these adeno-associated vectors. We expected again a decrease in the number of red cells, but this was not the case. So in our hands, we had a very inefficient, let's say, transduction of these astrocytes using these adeno-associated vectors. So we decided to move forward, changing strategies, and go directly into the brain of mice. And to do so, we screened the different types of adeno-associated vectors to see their distribution in the brain, and also to check for their specificity in targeting astrocytes. And we selected this AAV2 DJ8, it's a particular serotype. You see here in these vectors were expressing a green fluorescent protein in this case, and you can see here these are brain slices of a mouse that was injected as neonate with this vector in the brain ventricles. You see a nice expression of this green fluorescent protein along different regions of the brain, and also you see that a lot of these green cells are astrocytes, so meaning that we can target these astrocytes with these vectors. So we selected this serotype. And in parallel, we also characterized the mouse model of Alexander disease that recapitulates some hallmarks of the Alexander disease pathology, including the presence of these protein aggregates called rosenthal fibers, which are here in red pink, that are present in astrocytes labeled with GFAP. And we did a very careful, let's say, characterization in terms of time and region, let's say, checking the time dependent and then the region dependent appearance of these markers, so rosenthal fibers and the accumulation of GFAP, in order to establish some reliable readouts that could be useful then to monitor the efficacy of our therapy. So far, this data have been collected in the framework of this pilot project that was supported by ELA. So we have selected the tools, the reagents in vitro, and also we have identified the tools and the readouts in order to move on to the in vivo, let's say, experiments that we are currently starting. Thanks again to the support of ELA. So in this new project, the aim is really to test these gene editing approaches in the murine model of Alexander disease. So I finish here to acknowledging all the people in the lab that is mainly involved in the project, Vasco, Anna, Ilaria, and the other Ilaria, all the other people of the lab, the collaborators in our institute, and of course the funding agency and ELA first among them. Thank you for listening.
Thank you to you, Angela. This is another very impressive presentation that we hope can give a very quickly new possibility for the treatment of this very aggressive leukodystrophy. So we can move to the next speaker, that is Professor Peckney from the Clinical Neuroscience Department of the Godwell University, and you will talk about Alexander disease. So Professor Peckney, you can start your presentation. Welcome.
Good afternoon, everybody. We are getting the presentation on the screen, and here it is. I would like to start by saying thank you to ELA for organizing this important meeting between families, interested individuals, doctors, and researchers. I will try to...
Sorry, Professor Peckney, we have some problem with the volume of the audio. because I can't hear.
You don't hear me enough.
Okay, now it's
Is it,
perfect.
Actually, I will try again. Give me a second.
No, now we can hear you very well.
You can hear me well.
Yeah.
Perfect. Excellent. So those of you who didn't hear me from the beginning, again, thank you, Ella, for organizing this meeting. And I really appreciate that we are having many lay people today. And I will adjust that talk, my talk, in a corresponding way. And I would like to invite you first for a short journey through the most complex organ we know in the universe, and that is human brain. If we start with few numbers, we have about 10 to the power of 11 neurons, and a single neuron can connect and therefore communicate with up to 100,000 other neurons. We have about 1 order of magnitude high number of glial cells, and as you know, and as you also heard from previous speakers, these are astrocytes, microglia, oligodendrocytes. And if we think of astrocytes as single, astrocyte can connect and communicate with thousands of others via gap junctions, which are small openings in the membrane of these cells. Now, how does the brain work? Obviously, in my 20 minutes, I'm not going to give you any detailed account on that, but we should view this system as two extremely complex networks. You know, one of them you have all learned about at school, and that's the network of neurons. Neurons are electrically active cells in the brain, and we teach children at school already at the age of about 10 years that there are electrical impulses which are converted into chemical signals on a synapse. But then there is also another network in the brain, and that's the network of astrocytes, about which we know much less. But we have seen in the last 1 to 2 decades that astrocytes might be viewed more and more as the brain of the brain. Why? Because they control many functions of the neurons. They decide when and where neuronal synapses will be made, how they will function, and they also are responsible for modification of this neuronal network in the context of the disease. So sometimes our medical students ask whether a brain is a big supercomputer. And we always answer, we don't think so, because that analogy is not correct. First of all, at the moment there is no supercomputer available which would just come close to human brain, but you might say it's just a matter of time. But our brain, the hardware of the brain, is continuously changing. So as you listen to me, you are making billions of new connections in your brains. Most of them are transient, but hopefully some of them will actually stay and will undergo a process we call memory consolidation in medicine. So when you come home, you will get some take-home messages with you still, or when Monday comes, since many of you are at home at the moment, and you will remember my talk and all the other talks. So this plasticity potential is very important in health, and it's also very important in disease context. So astrocytes, you heard from a number of preceding speakers about, are among the most complex cells in the central nervous system, and all among the most complex cells in the body. They have thousands of cellular processes, they have huge heterogeneity, they extensively communicate with each other, and they form a network, as I already mentioned. And if you want to pause for a moment and to just think about that, a single astrocyte can access, and we now know that it means control, as many as 2 million synapses. So it's an immensely complex network. And if you look at individual astrocytes, you can think of them as crowns of trees, and everything else within these domains, and you have billions of them in your brain, Everything else is within that domain being accessed and controlled, such as neuronal synapses, microglial vessels, other components of the brain. So astrocytes do many things in the brain, and I don't have time to list them, but they are very much involved in the function of neuronal synapse, control of blood-brain barrier, they control blood flow. But when something bad happens in the brain, and it can be a neurodegenerative disease, stroke or trauma, astrocytes become reactive, and we talk about reactive gliosis. And in many cases this has been selected throughout evolution as a positive response. It helps to handle acute stress. It helps to limit the damage to the tissue or to parts of the brain. But in some situations it is maladaptive. It is actually not good, and then it's an important opportunity for treatment intervention. And Alexander disease, my topic of today, is a brilliant example. It's a problem which actually starts in astrocytes and which develops into a very, very detrimental, maladaptive function of these cells. So Alexander disease was the first monogenic disease known to actually originate in astrocytes. And there was a paper published in the Nature Genetics already 22 years ago, but it was such a milestone that I want to actually not only mention it, but to honor the first and the second author by showing the pictures. And I think that the gentleman on the left, the Professor Albi Messing, he is with us and listening today. The gentleman on the right is Michael Brenner, and they have found that mutation in the protein you have heard about. But because I don't like abbreviations that much, I will name it gliofibrillary acidic protein, or in short GFAP. It's a principal marker of a stress handling system of astrocytes, which is part of the cytoskeleton and which is known as intermediate filaments and sometimes also known as nanofilaments. Gliofibrillary acidic protein. And what Albi and Mike identified already more than 2 decades ago was that mutations in GFAP can lead to this very devastating disease. So as you already heard from Dr. Gritti, Alexander disease is a very rare but fatal in most cases, often very early onset leukodystrophy. And I underline this, leukodystrophy, because I want to emphasize to all of you that nature doesn't recognize stickers. Nature doesn't know the categories of the diseases. That's what us doctors use and researchers to really understand each other, to communicate. So by learning a mechanism of one of leukodystrophies or one of neurodegenerative diseases, it can help a great deal to understand the pathogenesis and to find out opportunities for treatment for other diseases. And it leads to massive loss of brain tissue, and you can see it even if you are not scientists or doctors, which is the case with most of you. You would guess that something is wrong in the when you look at the picture in the middle. And indeed, there is a devastating change which happens in the brain in Alexander disease in many, not all, but in many of the affected individuals. Of course, it leads to severe clinical consequences. At the moment, we have no treatment available. There is an ongoing clinical trial you have already heard about a little bit, and I would also say a few words about it. This disease is caused by a toxic gain of function of this protein, which you now know, gliofibrillary acidic protein. It leads to very prominent and widespread reactive gliosis, one of the most extensive reactive gliosis we actually know in any disease, and it also leads to characteristic accumulation of aggregates which contain GFAP but number of other proteins such as alpha-B crystallin, heat shock proteins, which actually every medical student and every doctor knows as Rosenthal fibers, and you can see them, and you actually saw them in one of the previous presentations, Dr. Gritti's presentation before. And that's something which you see very much on the right. Now, from a clinical perspective, we recognize infantile version and onset before 3 years of age, and that's something like 2/3 of patients. There are characteristic MRI changes, seizures, dementia, macroencephaly, epilepsy in many cases. Other signs, survival is for a few months to years. Juvenile onset, it's roughly 1/4 of the patients between 3 and 12 years of age. There would be ataxia, which means lack of coordination of muscular movements. There would be breathing difficulties, pyramidal tract signs, which would be in particular manifest as muscle stiffness, weakness, dementia. Survival is variable. And adult onset, which is only about 10% of the cases, and it's even more variable, sometimes might happen without cognitive involvement or epilepsy. But diagnosis is confirmed in all these cases by demonstrating mutations in GFAP gene. You already recognize the picture on the left. The upper picture on the right, you see those granular, dense objects. So these are the Rosenthal fibers, and we can zoom in, and at the level of electron microscopy, you will really see them as very characteristic aggregates. There are a number of animal models, but until a breakthrough finding or breakthrough publication just 2 years ago in Science Translation Medicine from the laboratory of L.B. Messing, most all of the models had substantial weaknesses. This has been substantially changed and improved by the rat model. If you look at the upper top panels, you can see on the left a normal rat brain, and on the right the rat brain from a rat with Alexander disease. You can see the cells are more dense. They look more dense. They are not more dense, but they actually express higher amounts of gliofibrillary acidic protein. You also see very prominent in the middle Rosenthal fibers. Hip means hippocampus, part of the brain. SC means spinal cord. So it's throughout the central nervous system. And as many of you would already guess, the lower panel gives you appearance of these characteristic landmarks at the level of electron microscopy. Now, the picture on the left looks very intimidating. I will make it friendly for you. So, first of all, these are 2, 3, 4 parts of the central nervous system. HC is hippocampus, cerebral cortex. CC, BC would be another part of the brain stem. SC would be spinal cord. These are proteins, 4 proteins which are astrocyte specific. We don't care at the moment about their names, but what we do care about is that WT, standing for wild type, means that they are very much expressed in those normal rats, but the R237, so these are the rats carrying a mutation in GFAP.
Sorry, but I have some problems with the audio again. I don't know if it's my problem or...
This is important. If I test it now, does it get better? Maybe okay. I'm moving too much, so I will be a bit more
maybe.
So and please Elena, let me know if there is a problem reappearing. So the bottom line is that the rats carrying Alexander disease mutation, they have dysregulation, in this case actually downregulation of quite a number of astrocyte related proteins, and they have number of functional problems. I give you one example. Clinically, a major problem connected with many diseases is swelling, brain edema, because astrocytes have very important role in the brain. They have a principal water channel, aquaporin 4, which is important for regulating water in the brain, the passage of water in the brain. And if you look at the bottom picture and look at the red symbols, you see that consistently the water content is increased. So it's an edema-like situation in Alexander disease rats compared to normal rats. So there are many signs in these rats which actually correspond to those seen in patients, and it is going to be very, very interesting model. The disease trial, which I will comment on in a few moments, also can be done in animals, and when those rats were given antisense oligonucleotides with the idea to decrease the expression of GFAP, it really worked. You can see it on the panel on the right. Well, let's start with the panel on the left. If you look on the left and compare F and G, you actually see what happens, the change in the readiness, which is the amount of GFAP, what happens after administration of antisense oligonucleotides. Body weight is a good parameter of success with an experimental therapy. And if you look at the middle panel on the right, you actually see that the Alexander disease mutant rats have lower body weight, and the Alexander disease mutant rats, they actually normalize their weight when antisense oligonucleotides are given, or they start to be given at 3 weeks of age, and it's also effective at 8 weeks of age. And you can also see on the right grip strength, which corresponds to another clinical parameter which can be quantitatively measured, and this therapy has improved it. Who are we? So what I am presenting this today to you is a consortium which we built together with ELA, with
Sorry,
another...
Professor Peckney, there is another problem with your audio. I don't know why. Maybe it's something related to the connection, Wi-Fi connection, or not.
You know, I
Okay.
am having rather good connection,
So
but
it's
I
a
don't...
problem of the
Better
microphone.
now? Is it better now?
Okay, yes.
But tell me if it's not, because what I will do is give me a moment. I will do this. Now I am trying to use completely different system. Is it better?
Yes, sure, it's much better.
Okay, so I have changed it very substantially. So I was just in the middle of introducing who we are, and
No, I'm sorry, but we have some problem. I don't know, maybe Elise can help us. It's a problem of my audio, or
exactly. Can
maybe.
would you please, somebody else, kindly tell me if the problem also comes to if the problem is general, because
Now I can hear you.
myself.
We
Why
hear
can't-
you with echo, but we do hear you.
Okay, thank you. So it's my problem.
Thank you very much. I will continue, and I will speak slowly so that the message gets across.
Okay.
So we are one of the 22 programs in Europe supported by EU as a joint program in rare diseases at the end of 2019, starting 2020. And we put this together, ELA Hall from Utrecht, myself from University of Gothenburg in Sweden, and we built a team of, as you can see here, 8 members. ELA, VKS, which is another Dutch patient organization. There is Professor Kubista's group from Prague, Itamar Harel's group from Hebrew University of Jerusalem from Israel, Dr. Henrik Alenius group from Lund from Sweden, and Professor Dolores Perez Sala laboratory from Madrid from Spain. Our name is Alexander because of Alexander disease, the astrocyte nanofilament system. Again, nanofilament is another term for intermediate filament, of which, of course, GFAP is the main player in astrocytes. The astrocyte nanofilament system in Alexander disease, from molecules to function, uncovering new leads for therapy. So what is our angle to Alexander disease? Well, we hypothesize that this negative maladaptive response of reactive astrocytes is a very principal part of the pathogenesis of Alexander disease, but that it's also highly relevant to other leukodystrophies and other brain diseases which have as a common denominator prominent astrocyte reactivity, prominent reactive gliosis. We also have a parallel smaller network funded by the Spanish La Caixa Astromed consortium headed by Professor Dolores Perez Sala, and jointly we aim to understand the maladaptive response of reactive astrocytes and of course to find out the cellular and molecular mechanisms which do lead to this pathology as an of course necessary foundation for designing future treatment strategies. The experimental models we use include the humanized models when we use induced pluripotent cell derived systems, and I will explain that in a few moments. We use this system from Alexander disease patients to generate and to study brain organoids, to study cell responses and the effect of Alexander disease mutation on astrocyte neuron co-cultures. And we are also building a killifish Alexander disease model as another experimental platform. The term iPS cells appeared already at this meeting a number of times, so I presume that all of you, even though you are lay persons, you know that these are induced pluripotent stem cells. The Nobel Prize in Physiology or Medicine in 2012 went to these two gentlemen who actually developed that system, and it means that you can isolate cells from blood or from skin and turn them into many different cell types. And the advantage is that you can build humanized systems in which you could study aspects of disease pathogenesis which might be more closely resembling human situation than some other experimental mammals. In other situations, of course, you have to use the experimental mammalian systems. But it's a very good setup also to identify novel treatment strategies. And if I show you this little video, and this is the system which is being used in Ellie Hall's lab, this is a brain organoid. So this is a very primitive brain-like structure which can be like half a centimeter, 1 centimeter large, and one can really model different stresses in it. One can derive it from patients with the respective diseases. One can have controls in which that mutation is corrected. And this is one of the systems which we are using. In our research. I will give you only one slide to summarize our data, and I would like to take you to the realm of something we call single cell gene expression profiling. This was a science fiction years back, but now it's an everyday reality, and it means that you can isolate cells from any organ or tissue you wish, or you can actually use cells from these induced pluripotent cells, and you can interrogate every single cell and get an idea about the genes which are being expressed. So that allows you to generate molecular definition of cell populations, to identify new cell types, their responses, both in a health situation and in a disease context. So what you see in panel A on the left are actually co-cultures between astrocytes and neurons, the blue ones derived from patients with corrected Alexander disease mutation, the red ones from non-corrected mutation. So you actually can see what happens when they talk to each other. And you can then get profiles for thousands of cells, and what you see on the right gives you an idea about how individual subpopulations of astrocytes can be defined. I'm not putting any genes there, this is just a scheme And you can see, just looking at the colors, green, precursors, meaning immature cells, blue, astrocytes, red, neurons. And if you look at this scheme here, you can all of you see that the green segment is much smaller here. This is a normal situation. This is the normal neurons, normal astrocytes. These are the Alexander disease astrocytes, normal neurons, and both of them Alexander disease. So this gives you an example that actually differentiation of cells in Alexander disease derived co-culture systems is very much affected. So indeed, that is one of our main findings, and of course we have a lot of molecular data on that the iPS cells derived from Alexander disease, they show impaired differentiation, and also they are more sensitive to various stresses. So these data are of course very much the foundation for designing new treatment approaches. And because we are talking treatment here, and you are all interested in treatment, I would like to say, as the very last slide, a few comments on the ongoing clinical trial, which is led by Ionis Pharmaceuticals, with LB Messing very much in charge, but actually a number of speakers you have heard today are attached to it in various ways. This is a phase 1-3 multicenter double-blind placebo-controlled studies. Participants are randomized in a 2-to-1 ratio. 2/3 receive the antisense oligonucleotides, 1/3 receives placebo for 60-week period, and then all the participants will receive the antisense oligonucleotides for additional 60 week. The patient range is wide, from 2 to 65 years of age, and the study is expected to be concluded at the end of 2025, and the primary outcome measure is a % change from baseline in the 10-meter walk test. And it's a quite invasive administration. The antisense oligonucleotides are administered by intrathecal injection once every 12 weeks until week 109, and so is placebo. And for additional reading or additional information, I would like to mention we just published a review with a similar title to my talk. And what I in particular would like to mention are these Alexander disease research updates, which is a fantastic work which LB Messing is engaged in, and this is the connection. So you can actually listen to, in a podcast, to all interesting updates from the research, and sometimes even from clinical fronts when it comes to Alexander disease. With this, I would like to thank you all for listening, and those of you who do that, for supporting research on neurological diseases. Thank you very much.
Thank you to you, Professor Pecny, for this incredible journey into the brain and astrocyte biology. I know that you are a world-leading expert in astrocyte biology and reactivity and Alexander disease. So it's my pleasure to open all the session for the discussion. I want to thank all the presentators, all the speakers, and a little comment, if I can do, is that all these presentations, the level of the presentation is amazing. And if I compare these studies with those that were presented years ago, I have to realize that a lot of progress have been made in the particularly in the molecular mechanism of this disease, and also in the generating trials and possible therapeutic strategies. And it's a very important, and I have to thank the ELA for this, for all the work that they have done in supporting the research. And as you know, rare disease very difficult to be supported, and there are there are no many possibilities to have funding. And thanks to this organization, now we are able to have this kind of presentation that indicates that the roads towards the therapy is open, and patients have the hope to be treated in the next year. So I think that we can start with the discussion. If there are from the audience some questions.
So you are all invited to raise your hand if you'd like to ask a question. We can open your microphone. At the bottom of your screen, if you'd like, you can select the hand. There's no hand right now.
No, I think there are some questions in the chat, but one is in Spanish, maybe.
I think I answered it.
Ah, okay. So, okay. It doesn't appear your answer, so it seems no problem. Okay. So please don't be shy. We can answer in the chat if you want, if you prefer. So there are 75 participants, so not so it's a considerable number of participants. Maybe Elise, what do you suggest?
I don't see any hands risen. So, there is no question from the audience at this point. Ah, here it comes. There is one. So I'll just give you the possibility. Alicia, if you'd like to ask your question.
Alicia, we can't hear you.
Hola, buenas tardes, gracias a todos. Hablo desde España, hablo en español, supongo que hay traductor. Quería saber para realmente todas estas terapias que se están viendo y que se están haciendo, todas las terapias genéticas, ¿cuándo llegarán realmente al mercado? ¿Cuándo estarán a disposición de los enfermos que padecen alguna de estas enfermedades? Muchas gracias.
Maybe Professor van der Knapen-Tekny or Dr. Angela Gritti want to give an answer?
Can somebody translate it for me?
Mm-hmm.
I didn't get the question. My Spanish is 0.
The question was when the gene therapy or therapeutic strategies in trials will be available for patients.
Dr. van der Knap, if you'd like to hear the translation, you can use the English channel of the translation as well. Then the question will be translated.
Okay, I don't see it, but it doesn't matter.
Timing for therapy is not so easy to know in advance.
Maybe I can make a brief comment, if I may.
Yeah, yes, sure.
That's a most appropriate question. Thank you. We must realize that apart from the clinical trial, which I commented on, all the other lines and avenues, and you heard so nicely from Dr. Gritti about one of them, they are in the experimental stage. So the new therapy can only happen after the clinical trials are evaluated. So when it comes to Alexander disease specifically, then of course the clinical trial will likely be fully evaluated somewhere between 2025-2026. The frustration from the slow pace here is understandable. There is very limited funding, and once more I really would like to congratulate LB Messing, who is here with us today, for what they did with that limiting funding and what we are all trying to do. And I think that Mario might comment in a similar way for the other or on other diseases. I just made a statement for Alexander disease, but it's probably very similar when it comes to the related areas?
Well, I'm not sure. Thank you so much. I'm not sure I quite know what to say. I think that for several diseases, drug therapy is really developing, and that's also something that.
Acabo de preguntar, soy la única que pregunta.
We have some problem.
Okay. Shall I go ahead?
Yes, please
Yes.
go ahead, Dr. Van.
And in Amsterdam, we are also working on gene therapy, and like we published, we had serious off-target effects actually in the mice. So the mice became worse when we tried to do gene therapy for vanishing white matter. And so I think that there are still safety issues. You don't want to make a patient worse. And then for some diseases, it is, the diseases are devastated by itself. But like a disease like MLC is a very slow disease. So if gene therapy would make it worse, I would be very worried. So right now, we are also working on gene therapy, but right now I'm more optimistic about the speed of drug treatment. I see Elena nodding. That's
Yes,
something
I'm nodding.
that's easier.
But for MLC, it's different because, as you have said, is a slow disease. So there is the window for time for therapy, and it's quite different. So we have some question in the chat. I don't know if I have to read them or the researcher can answer by writing the message. For example, for Dr. Meneghini and Gritti, what's the timeline for the mouse study? When do you think you will be able to know if the adenoviral vectors works in mice? I don't know if Angela, here they are.
Oh, yes. So the timeline for the mouse studies, I mean, as I mentioned, there is this new project that we are starting, then Vasco maybe can add something. The timeline to have this mouse studies completely is about maybe 2 years, let's say, but in order to have some ideas of the efficacy of the treatment. In this case, is not really, well, the adeno-associated virus is just, the vehicle to deliver these gene editing machines. And of course, this is an additional complication because the real, let's say, effector is the gene editing machinery, but then we have to use the viruses in order to get this machinery in the brain tissue. So another possibility, of course, that we are thinking, not only our group, but in the world, is to not to use vectors, not to use adeno-associated vectors, but to use the sort of lipid nanoparticles or something which is not viral in order to, let's say, avoid the potential problems that the vectors could also give. So there are different layers, let's say, of complication with this these approaches, which have to be very carefully evaluated, as Dr. Van den App was saying. Of course, a pharmacological treatment, it's easier in a way because you can always withdraw if you see some side effects, but of course this is not possible with gene therapy. So you have to be very, very careful, and you have to have a lot of controls and safety checks, let's say, in order to proceed, because these are different from drugs, okay? So these are quite more complicated systems, but the potential advantage in the future, so if everything is, turns out to be reasonably safe, I mean, we don't never have 100% of certainty, that could be a one-shot treatment as compared to the pharmacological treatments or the antisense oligonucleotides, in which you have to give this treatment for the lifetime, basically.
So I think that this answer is good also for Mr. Martini that asked about the timing for the CRISPR-Cas9 strategy to fix the genetic mutation. And I want to read a message by Simone Boria Lauro. And this message is the following. I have no question. I only want to thank the researchers about Alexander disease. My daughter died 14 years ago of this disease. I'm happy that others' parents will not suffer what we suffered. So I would like to read this message for all the audience. And there are also other two questions for Professor Peckney on Alexander disease, but I think that you can answer by writing a message, or do you want to answer about gene therapy of Alexander disease in USA? But Dr. Vasco Meneghini is writing an answer. And okay, there are also messages that want to thank the researcher work, and is a mother of an MLC patient. And there is a question in French, maybe on Canavan disease. I can try to translate. It's about treatment in course in United States with Dr. Paola Leone. They want to know if results are satisfying and if you know about this treatment. Maybe Dr. Eckhart can help.
Yeah, I know this trial, and currently I'm not so familiar with the details. I think there are seems to be some promising data on this as far as this is published, but I cannot definitely say whether how promising these data are and how long it will take for therapy.
Okay, maybe there is Mr. Wagner asking Professor Peckney about the azotherapy. Maybe Professor Peckney is writing an answer. So
yes, I was writing the answer, and it's fair enough to mention that I didn't show any specific data related to differences in individual molecular pathways because we are still in the middle of this analysis. So there might be potential targets related to the stress responses, but this work has to be finished first, and of course then such targets would first be validated in respective animal models before one can even think of a clinical study. So it's not one step before, but it's fair enough to say it's 3 or 4 steps before.
Okay, thank you. There is a general question about guanabets. There is Miss Sara Siciliano who is asking if we can have more information about the type of drugs about guanabets for the treatment of vanishing white matter.
Okay, meer information. The trial is registered on the European trial website. So if people want to read it, then I would have to look up the website. But it's the European Registry for Trials. That's one. And another person was asking me whether patients can have access to the website, to the drug outside the trial, and the answer is no. Guanabens is not, human quality guanabens is not available. So that means that we had it made specifically for the trial. Patients from apart from the clinical entry criteria, it says reasonable travel distance, which is of course sort of a stretchable inclusion criteria. And patients from like Western Europe, if they fulfill the inclusion criteria, they can just come, and the trial covers all expenses. So it covers travel expenses and also everything else. Does that answer your question?
I think yes, but maybe she wants to know which type of drugs, which.
It's an alpha-2 adrenergic drug. So yeah, and I would also, Elise, be happy if you would share my email address or the Vanishing White Matter registry email address, because I think
we will, and also we'll have an opportunity to include it in your follow-up article that we'll present to the families.
Because then the most families, of course, want to know, does my child qualify for the trial? And that's difficult to answer in general, because we would have to look at the clinical details. But we are always answering our emails, and so we are happy to receive emails with questions on the Guadalupe trial.
Absolutely.
Okay, I think that there are no more question, or what do you?
I think the time is up.
Okay. We have only 3 minutes. We are only 3 minutes late, so
it's perfect.
It's perfect. Thank you again very much to all of you, and it's been very important for us to share our results with all the ELA families and parents and patients. So thank you again very much, and we hope to see you next in the next meeting. Bye.
Thank you very much. We'll have a short 15-minute breaks, and we'll move on to the next session. Thank you, Dr. Ambrosini, and all our speakers.
Thank you to you.
Thank you.
Thank you.
Thank you. Thank you.
Welcome back, everyone, for this second part, the second session for today. We have the pleasure to have 4 guests with us this afternoon, and we will start with Dr. Marie-Louise Frémont from France, who will tell us about the follow-up of an ELA-funded trial in Aicardi-Gouttière syndrome. Please, Dr. Frémont. We cannot hear you.
Yes, hello, everyone. Bonjour à tous. So I will speak in English today. So I'm very pleased and honored to share with you this moment and our follow-up of an ELA on the trial in Aicardi-Gouttière syndrome. So I am Marie-Louise Frémont. I work as a pediatrician at Necker Hospital, and I work as a researcher in Yannick Rowe laboratory in Imaging Institute. So I was very lucky to did my PhD with Yannick, and now I'm still working with him. So today, we are going to speak about Aicardi syndrome. So as you may know, this disease is including type 1 encephalopathy. So these diseases are monogenic disease, include in inborn error of immunity. And their definition is that they are disease associated with a constitutive type 1 interferon signaling, and that the concept coined by Yannick in 2011 include that this constitutive type 1 interferon signaling is directly relevant to the pathogenesis. And this concept also support the hypothesis that targeted therapy should alleviate the symptoms. And actually, Aicardi syndrome has been described almost 40 years ago at Necker Hospital by 2 French neuropediatricians, as you may know. So Françoise Gouttière and Jean Aicardi, who described in Siglin a very severe encephalopathy with intracerebral calcification, as you can see here on the CT scan. And they also observed in the patient severe chilblain, what we usually observed in patients with lupus. And so already at that time, they made a hypothesis that this disease was a monogenic disease, but at that time they didn't find any mutation associated with this disease. And as you may all know, Aicardi-Goutieres syndrome is mainly affecting the brain with severe encephalopathy. The patient present mainly with diplegia spastic, dystonia. Their brain MRI displayed a severe white matter disease, and we can also observe intercranial calcification in the brain MRI or CT scan. The second major organ affected in this severe is the skin, with this chilblain that can be very disabling for the patient, especially in the winter when the lesions are worse. But this disease actually can affect most of the organ. Some patients have recurrent fever, short stature, they can have autoimmunity up to lupus with all the criteria for the diagnosis of lupus. Other organ involvement can affect the heart, the kidney, the endocrine organs. So I would say that although this disease mainly affect the brain and the skin, we are learning with the patient, the families, and all the literature that this disease can affect almost all the organs. So I told you that the disease was described in 40 years ago, and Yannick Rowe discovered the first mutation associated with Aicardi-Goutieres syndrome. That was quite a long time ago too, in 2006. So he described mutation affected TREX1. So what is TREX1? TREX1 is a nuclease that in the cytosol remain the double-stranded DNA. So this is basically the activity of TREX1. And when TREX1 is affected by a mutation, TREX1 is not working anymore, and there is an accumulation of double-stranded DNA in the cytosol of the cells. And that was also observed in patient cells, where there was, here are the cells from the patient, here from the healthy control. And you can see that in the healthy control, you do have a release of nucleic acid, but in the patient, there is no nucleotide released in an assay to assess the activity of TREX1. And when there is this accumulation of DNA in the cytosol, the cell activates a pathway that usually is there to detect foreign DNA coming from either a virus or a bacteria. And so the cell triggers this pathway that we call cGAS-STING pathway. And this leads to a major production of interferon. And this leads to the constitutive activation of this pathway in patients, but without any virus or bacteria infection. And we do believe that this excess of interferon and all the proteins that are produced because of this excess interferon leads to the tissue damage that we observed in patients. So now, some years later, we know now 9 genes that are mutated in Aicardi-Goutieres syndrome. And if we talk about all the type 1 interferonopathies, there are more than 30 genotypes that are now associated with this disease. If we focus on AGS, all the genes involved have a direct or indirect role in what we call nucleic acid. So nucleic acid can be DNA, as we have seen in the example of TREX1 mutation, but nucleic acid can also be RNA. And these 5 specific genes, TREX1, RNase H, B, C, and SAMEG1, they do have a direct role on the metabolism of nucleic acid, either removing them from the cytosol like TREX1 do, either having other specific function that I will not detail today. And so the major question for us as a scientist was, what is the source of nucleic acid? Because I told you the nucleic acid that are detected by TREX1 in the case that are not removed by TREX1 in the case of a mutation are not coming from a virus or a bacteria. And actually, we know that the human genome is mostly composed from non-coding DNA. So if you've got here the circle representing all the human genome, there is only a few parts in blue that are genes encoding protein that do have a role in human cells, and the other one, the other part, the major part, is not coding anything. And in that part of things that are not coding anything useful for our cells, our human cells, there is what we call the endogenous retroelements. They are 40% of the human genome, and they are derived from fragments of virus that have been integrated in our genome during evolution. So this has been with us for a long time, and so there are different type of retroelements. I'm not going to go in too much detail, but what is important to know is that it is that some of these retroelements have the ability of replication, so they can replicate and they can create DNA, nucleic acid in the cells. And to do that, they encode the protein they need to do that, that is the inverse transcriptase. And you've got here the picture of HIV and some pills. Why? It's because HIV, as you may know, is a retrovirus. And the tree therapy that we use to treat HIV patients are inhibitors of the inverse transcriptase that we call retrotranscriptase inhibitors. And so so with the idea that retroelements was the source of nucleic acid that were detected in the context of TREX1 mutation, the group of Murtry did a very elegant study already 6 years ago. They used human stem cells coming from a TREX1 Aicardi-Gouttière syndrome patient, and also cells from a healthy control. And they differentiate them in neurons, astrocytes, and progenitors. And then they do what we call an immunofluorescence, and they look at the cells. So in blue, you've got the nuclei of the cells. In green, you've got other parts that are important for the cytoskeleton of the cells. And they used a yellow marker to mark DNA that were not in the nuclei. And you can see on the screen, or you can believe me, but there are dots, yellow dots that were outside the nuclei and that were DNA. And when they treated the cells in vitro with an inhibitor of the retrotranscriptase, the dots disappeared. And when they used on the last panel a placebo, there were still the dots. So this was the first in vivo proof that indeed the retroelements were probably the source of nucleic acid that were not removed by TREX1 in the cytosol in the context of TREX1 mutation. And this can be also the case for the other genotypes in Aicardi-Gouttière that I mentioned to you, so RNase HBC and SAME-G1. So with this in mind, Yannick Rowe and Stéphane Blanche set a first trial in Aicardi-Gouttière syndrome that was funded by ELA, using a retrotranscriptase inhibitor in Aicardi-Gouttière syndrome, with the hypothesis that this drug would decrease the pool of retroelements in the cytosol of the cells of the patient, and thereby decrease the overactivation of the interferon pathway. So this study has been conducted in 2015 and the years after, published in 2018. So 11 patients were recruited, 8 finished the study, and these patients were treated during 12 months with 3 inhibitors of the retrotranscriptase. And what has been shown is that the interferon markers, either in the blood or in the cerebrospinal fluid of the patient, were decreased upon treatment. I will go with you in the figure. So here in this panel, we looked at interferon score in the blood, and you can see that under treatment, the interferon score decreased, and when the drugs were stopped, it increased again. These 2 panels, E and F, were performed in the cerebrospinal fluid of the patient, and you see that interferon protein or activity decreased in the CSF of the patient. So that was a proof of concept that indeed this drug were able to decrease interferon signaling in patient blood or cerebrospinal fluid. And this study also include the assessment of brain MRI in some patients. And interestingly, it was observed an increase of the cerebral flow in 3 out of 5 patients. So this study conduct was a proof of concept. The idea was not to assess clinical efficacy of the drug, but more the biological efficacy of the drug on interferon signaling. I must say that one of the limitations of this study was the taste of the drug and also the volume of the drug that were taken orally, and this has been a problem for almost all patients. So it was difficult for them to take the drug. So we are now following with this hypothesis and the wish to more assess the efficacy of retrotranscriptase inhibitor in Aicardi-Gouttières syndrome. And we are now a center for another study with transposon therapeutic as a sponsor. So the idea is the same, is to use a retrotranscriptase inhibitor in Aicardi-Gouttières syndrome and to assess efficacy on interferon signaling, but also of course, tolerance of the treatment. This drug is called TPN-101. It is an analog of another RTI, and there are quite a lot of in vitro data showing its efficacy potentially higher than the usual retrotranscriptase inhibitor, and also very convincing results from a phase 2B study in an HIV patient with a good tolerance. So we have the hope that this treatment would be well tolerated and efficacious in the patient. And apparently the taste is better and it's not difficult to take the drug, but this we need to see if it is real or if it's just what the sponsors say. So this is an European study. There will be 3 centers in Italy, 1 in UK. Our center in France is open for recruiting. And there will be 4 successive cohorts in this study. And the first cohort is only for adult patients, so above 18, and only for the 5 genotypes that I mentioned to you, so TREX1, RNase H, B, C, and SAMHD1. So what are the other perspectives for the treatment of Aicardi-Gouttières syndrome? I must say that there is an important effort in our group, but also in Europe and in the United States to ameliorate the treatment of this severe disease. One thing that seems to be mandatory is to have a rapid and earlier genetic diagnosis with better biomarkers, easy access for everyone to accurate genetic testing. What can we envision for treatment? Maybe a combination therapy with the RTI that I presented to you today, knowing that we observe a promising effect of JAK inhibitors on skin vasculopathy. I don't have time today to go into details, but we are not so confident that JAK inhibitors do have an effect on the brain, but this needs to be confirmed in further study. Another thing that we could envision is to deliver the therapy directly into the cerebrospinal fluid, because so far we are giving drugs that circulate in the blood, but we know from our experience that, for example, JAK inhibitors only pass 10% into the cerebrospinal fluid. And we are also very interested that what is being used in lupus, adult lupus patient, like the anifrolumab, that is a monoclonal antibody directly targeting the interferon receptor. This drug has been approved by the FDA and also by the EMA. So far, we do not have access to this drug for a pediatric patient, either with lupus or monogenic disease such as Aicardi-Gouttières syndrome. But this is also something that we're hoping to be able to try on in patients. And more in the future, there is a lot of in vitro work on small inhibitors targeting other component of the SIGA-STING pathway, such as STING inhibitors. So with this, I want really to thank you for your attention, and I will answer your question at the end of the session. I would like to thank Jan Nicro, that is really a mentor for me, and all the CROLab. So the CROLab is situated in Paris and in Edinburgh. I put you a picture of some of the people that work work on interferonopathies, and some of them are working on Aicardi-Gouttières syndrome. At the Necker Hospital, I want especially to thank Bénédicte Neven and Marie-Julie, that are a really important person that have a great experience in Aicardi-Gouttières syndrome and conducting all the clinical trial for these patients. Thank you.
Thank you very much, Dr. Freeman, for your presentation. Very encouraging. We will indeed take the questions at the end of the session. So please stay with us. And if you have questions you'd like to ask in writing, that is also possible. Experts might eventually just answer them directly in writing, or we can address them later orally. Okay. Our second speaker today will be Dr. Englund. The program was announcing Dr. Femke Klauer, but she cannot be with us today. So Dr. Englund kindly agreed to be our next speaker, and he will tell us about the Zellweger spectrum disease.
Well, thank you very much for the introduction. I see my screen still says Femke Klauer, but yeah, she's still on maternity leave, so I'm happy to fill in for her. Let me share my screen. So I hope you can see the presentation now.
So we can see your slides, yes.
Excellent. Then I will go to presentation mode. There we go. Okay. So I've been asked to present on Zellweger spectrum disorders. I'm a neurologist. I work at the Amsterdam University Medical Centers and at the Amsterdam Leukodystrophy Center, and we see quite a few children and also more and more adults with the Zellweger spectrum disorder. And I'm going to walk you through the characteristics of the disorder and the recommended follow-up. So what I'm going to be talk about is the brief biochemical background of the disorder, and then the most important clinical features as we see them today, and then with the recommended follow-up and supportive care that we offer. So peroxisomes were discovered almost about 70 years ago as small cellular organelles. on this nice immunofluorescence imaging, they're the little green dots. But they were mostly ignored for many decades, considered not very important. And only when they were linked to human disease in the 1970s did people become interested in them. And it turned out that they are very essential for normal cell functioning. So
I'm sorry, Dr. Anglin, we do not see the slides changing. We still have the non-full screen view of your
Oh,
slides.
excuse me. I'm going to try again. Excuse me. I have too many screens attached to my computer, so I'm going to try again. I'm going to move it.
Yes. Thank
Is
you.
it working now?
Yes, I think so. On the third one, yeah.
So right, so we go to the functions of the peroxisome. So if everything is okay, you should see a diagram of a peroxisome, and now it's known that peroxisomes are incredibly essential for all kinds of functions. They're involved in degrading certain components like very long chain fatty acids, but they're also important in making cellular components. Such as complex lipids, and are important in bile acid synthesis. So if you see all the biochemical processes they're involved in, it is not very surprising that they're implicated in human disease. So how do we classify? It's important to know how to classify. Peroxisomes have many functions, and it means that if there is a defect in a specific pathway, that usually results in problems. And because there are now so many different peroxisomal disorders, people have been trying to make it more systematic. And when we talk about peroxisomal disorders, we now talk about single enzyme deficiencies if there is only one specific pathway involved. So one enzyme lacking. But what we're going to talk about today are the Zellweger spectrum disorders, and they are characterized by the complete lack or dysfunction of peroxisomes. And this group is called peroxisome biogenesis disorders. So there are no peroxisomes. And how does that occur? Well, for the formation of peroxisomes in the cell, there's a category of proteins called PEXs, peroxins. And there are proteins that are part of the peroxisomal membrane, and they are involved in forming normal peroxisomes or importing enzymes into the peroxisomes. And if defects in these PEX genes arise, you get cells with no or very few peroxisomes. So in this little green image here, every little speckle is a peroxisome. And in somebody with Zellweger spectrum disorders, you can see that there's just this green background coloring, but with almost or no discernible peroxisomes. So these cells lack all peroxisomal function. And that affects all kinds of cell processes. So you get a shortage of some components, and you get accumulation of some metabolites that can be broken down. And this pattern of abnormal metabolites can be used for diagnosis, as we will see a little bit later. So it took almost 25 years after the description of the peroxisomes in the 1970s before it was realized that these peroxisomes could actually cause human disease. The first peroxisomal disease had already been described in the 1960s, but at that time the genetic and biochemical background was not understood. And it was called Zellweger syndrome after the person who described it. And he described very sick babies with characteristic facial dysmorphisms, abnormal brain structure, severe liver dysfunction, and also renal cysts. And they called it cerebrohepatorenal syndrome. Then it became very quiet. Later it was described in liver biopsies of children suffering from this disorder that there were no peroxisomes. And that's when everything went faster. So these babies had abnormal brain structure, mostly because the cerebral cortex was not normally developed, and there were deep cysts next to the ventricular system. And if you look at it in more detail, We all have these convolutions called gyri, and in people with Zellweger syndrome, very often, the severe Zellweger syndrome, these gyri look abnormal. They are smaller and there are more of them. And the classical very sick babies are very floppy, have feeding difficulties, respiratory problems, and a very severely shortened lifespan. But then again, the years after that, there were also babies who shared characteristics like poor vision, poor hearing, developmental delay, but with, in general, much better development. With much later presentation. And at first, all these disorders were considered distinct. So first there was the Zellweger syndrome, but children who presented later, they were said to suffer from neonatal adrenoleukodystrophy or from infantile Refsum disease. So the less severe cases got a different name, and now we think that is not very helpful. Now we consider them all part of the same Zellweger spectrum. So because they are biochemically and genetically overlapping, we now say Zellweger spectrum disorders, and all the old names have been abandoned in the research literature. So this is how we view it today. We say that Zellweger is indeed a spectrum of disease severity, and generally the earlier after birth symptoms occur, the more severe the disease. So the babies who present immediately at neonatal age are very severe. They have epileptic seizures, brain abnormalities, very hypotonic needs, gastric feeding tubes, have liver failure, and unfortunately a very short lifespan. And the later the presentation is, the better they do. So children present with also abnormal development, but less. And sometimes now we even pick up adolescents of adults with some neurological dysfunction, but with reasonable functioning, and sometimes even people with pretty normal functioning. And the only thing that all patients seem to share is the sensorineural hearing deficit, so the deafness, the abnormalities of the retina, and sometimes teeth abnormalities that everybody has. But the neurological and liver symptoms depend on the age of presentation. So this is what we think the spectrum looks like. So the prognosis depends, of course, on the disease severity, and the disease severity is strongly correlated to the age of presentation. And the classic Zellweger syndrome is unfortunately a very severe disease. And often children do not survive the first or second year of life and generally die of respiratory failure. Patients that present later often, and it says here, can live into adulthood, but actually most often live into adulthood, although those that present usually at early age below 4 are severely intellectually impaired, have many disabilities, but survive into adulthood with supportive care. And there's even some rare patients that present later that have completely normal intellect, especially the PEX ones. Yeah, it's always considered a progressive disease, and I suppose it is, but it is really slow. The people we follow in clinic, they get worse, but they get worse over many years or sometimes even decades, so it is not that progressive. Now, the diagnosis these days can, of course, if it's suspected on basis of clinical findings, it's easy to screen for all the peroxisomal metabolites. So the bile acid intermediates, the very long chain fatty acids, etc. Or, of course, do genetic analysis. But more and more in modern times, people get whole exome sequencing, so the complete genetic sequencing, if there is a developmental delay. So people are now much more discovered often. So we get more referrals. And sometimes it is needed to do functional testing if the genetic variants are of unclear significance. So as we've heard often today, unfortunately, there is no curative treatment for this disorder. But good supportive care really makes a difference. And because we kind of know the natural history now, you know what to look for. And follow-up can prevent some treatable complications at least. So for these patients, it is beneficial to go to I suppose, larger hospital with a multidisciplinary care team to receive optimal supportive care, also by rehabilitation, medicine, etc. And because Now, most patients actually survive into adulthood or even late adulthood. It's also important to think about transitioning to adult clinics because the care adults need is of the adults with Zellweger spectrum still need supportive care, different from the children. And we generally see children twice a year for monitoring, and because the older patients are much more stable and progress very slowly, after that, a yearly visit is usually sufficient. So what do you look for in patients with a Zellweger spectrum disorder? Well, because there is a problem with forming bile acids, there's a problem, there can be and often is a problem with fat absorption, and therefore also with the absorption of fat-soluble vitamins. So that those are vitamin A, vitamin D, vitamin E, and vitamin K. So deficiencies of these vitamins are very common in patients with a Zellweger spectrum disorder. And if they are deficient, if you test that, you can supplement it. And especially low vitamin K levels are dangerous because they cause vitamin K dependent clotting disorders. And we have seen over the years some pretty severe bleeding incidents in patients with Zellweger spectrum disorders that we're unaware of the diagnosis. So that is something that can really make a difference. So we generally advise to do a laboratory checkup for the status of the coagulation yearly and supplement with very high dosage vitamin K as necessary while monitoring the coagulation. So dietary management. In the old days, and I mean the 90s, patient often got all kinds of suppletions for essential fatty acids. It turns out that it is probably not beneficial and not needed. Same for phytanic acid restriction. Phytanic acid is degraded, is a peroxisomal function, and it is a dietary product. So like in Refsum's disease, people can accumulate, and it could be it could accumulate. We see that in Zellweger patients, the accumulation is usually very minor. So generally, you can check it once every few years, but it's generally not really necessary to follow any kind of diet. What is a problem, especially in the patients at the severe end of the spectrum, is poor feeding. And many of the more severely affected patients will need gastrostomy tubes, which generally increase well-being quite a bit for the caregivers and also for the children and adults themselves, because they no longer have to struggle to maintain weight. So that is something that we'd rather do early than late. Something that has been known for a long time, but rediscovered recently, is that we think about a third of the Zellweger spectrum disorder patients develop adrenal failure. And adrenals produce important hormones, mostly cortisol. And if you become cortisol deficient, that can lead to very severe complications in situation of bodily stress, like fevers or trauma. And it is clinically very difficult to recognize. Patients can be asymptomatic for a very long period of time, or only have very, aspecific symptoms like fatigue or bruising. And so what we also do is we recommend testing yearly or twice yearly for adrenal function, because if adrenal dysfunction is present, it is easily treatable with hydrocortisone, with synthetic cortisol. And that is also something that can generally really benefit quality of life. So another thing to be aware of is peroxisomes are also important in oxalate metabolism. And people with Zellweger spectrum can, not always, but can have a very high oxalate in the urine. And it can form crystals, and therefore can form kidney stones, which can be incredibly painful. And because many of these patients have severe intellectual disability, it's very difficult for them to tell you what they are suffering from. And it sometimes takes a very long time before then it is discovered that they actually have kidney stones. So we kind of monitor for that. It's recommended to monitor. And in patients with high oxalates, you can acidify the urine with potassium citrate, and that can prevent kidney stones. So it's relatively rare, but if it occurs, it's really painful and unpleasant. So this is something that that is also good to monitor and treat. So the liver disease, actually, probably the liver disease, the cirrhosis and the complications of that, like severe hemorrhaging and things like that, and liver failure, is probably what is in the older patients the most frequent cause of death. It's important to be aware of also in the older patients. And it is also advised to screen for sclerosis and involve a gastroenterologist if necessary to treat the complications of cirrhosis. Again, it is something that is very slow, especially older patients. Liver disease is often relatively stable, but it is something that can also cause a lot of symptoms if not preventively managed. Now, other complications that occur, patients with Zellweger spectrum can develop a leukodystrophy, can basically occur at almost every age. It can occur in young children, but also in adults. It's unknown how many patients eventually get it. Probably for the most common PAX mutations, the majority will not get a leukodystrophy, but it does occur. We don't routinely screen with MRI scans because it has no treatment implications. This is a leukodystrophy we cannot treat. And because in many patients with intellectual impairments, that would require MRI scanning under sedation every time. So, but it is something to be aware of if somebody with Zellweger, which, like I said, is generally a very slow disease, often rapidly shows deterioration in neurological function. So very severe psychiatric symptoms, cognitive decline, rapid decline in motor function with problems swallowing or walking, then it is probably a good idea to do an MRI scan, because if you find the leukodystrophy, that has prognostic implications, because that generally means that that somebody will have very severe complications in the near future, which is important for counseling and arranging care. What is typical of the leukodystrophy, the supratentorial lesions, and the white areas, very aspecific, but what is quite specific is the region in the helix of the nucleus dentatus in the cerebellum is pretty typical in this pattern that is often seen. So one thing, that there is no curative treatment at this time, as we mentioned. It has often been suggested that suppletion of cholic acids, so that is a bile acid, could be beneficial. There are indeed some trials that show that it has a biochemical effect. It decreases the bile acid intermediate, so there's a biochemical effect. It does something. Only it has never been shown that it really has an effect on well-being, and it does have complications. Bile acid therapy, especially in older patients, they often have abdominal complaints and diarrhea. So it is not something that we would recommend in routine care. And it is something that requires better trials before it is considered, could be considered as a standard treatment. So to summarize, Zellweger spectrum disease is a very complex disorder which affects many organ systems. People have many medical problems generally, so a coordinator of care is very important, and it can be a clinical research coordinator or a nurse, but something that can guide families through all the complex appointments and tests in the hospital. And there should be a single physician that also coordinates care. It can be a neurologist or a metabolic specialist. It kind of depends on the specific healthcare system that is organized differently in every country, but usually as needed, different specialists should be involved. Like people with prominent liver disease will need a specialist for that. Generally, you also need an eye doctor, ear, nose, throat specialist. Dentists are very important because people have complex tooth problems, etc. And visits pediatric patients a little more frequent, and older patients are generally much more stable. So in adults, we sometimes do once in every 2 years, which seems to be enough. So conclusion, I think that has been discussed many times today. Unfortunately, in this day and age, we can diagnose many genetic disorders, but we cannot cure many. But that does not mean, the fact that we don't cure does not mean that we can't do anything. Care at this point is probably just as important. And also for disorders like this, I mean, it doesn't stop at the diagnosis. There are many complications that you can treat and that you can make better. So you should have these patients come in so you can help with the complications that occur. Yeah, and for the future, and that is something that we've heard a lot also today, it's very important, I think, to centralize care so that we learn more. And once we advance, that we have access to the patients to also try the new therapies that will definitely evolve in the coming decades. So with that, I would like to close. And if anybody has questions or suggestions, please put them in the chat. Thank you very much.
Thank you very much, Dr. England, for this thorough presentation on the Zellweger spectrum. Our next presentation will be a dual presentation from Dr. Marco Cecchini and Andrea Del Grosso on our first talk on nanomedicine and autophagy modulation in the mouse model of Krabbe disease and the work they've been conducting with ELA.
Okay, hello, thank you for the introduction. Can you hear me? Can you hear me?
Yes, we can hear you.
Okay, great. Now I share my screen. One moment. Seems it doesn't allow me to share the desktop. Just a moment.
How is it going on? If you...
I should exit the Zoom and then reopen the program. Just give me a couple of seconds.
Okay, otherwise you could maybe send it to us. We can see if we can share it from our side.
Just let me just try.
Okay. All right, so we'll wait a few moments. The connection of Dr. Cecchini and hopefully reconnection, and hopefully the presentation will start soon. Thank you for your patience. So don't go anywhere. We're still waiting for Dr. Cecchini. back with us. And hopefully we'll start in a few moments. We apologize for the technical issue. We hope it will be resolved soon. Dr. Sacchini could not share his slides and had to step out, but he should be back with us shortly. And hopefully we can start his presentation soon. Okay, he's back.
Okay, sorry for this. Okay, I go to the presentation mode. Okay, can you see it in presentation mode?
Yeah. Yes, we can see it. Thank you.
Let's start and okay, so thank you for the presentation. Thank you, organizers, for having invited us to present to the families our research activity. This talk is divided into two parts. In the first part, I will introduce crab disease and provide an overview of the therapeutic strategy, which is based on nanomedicine. The second part is dedicated to a second project in which we studied autophagy modulation as a potential complementary therapy. Ambra Del Grosso, who has been working with me in this field for many years, will present these latter results and conclude the seminar. Crab disease is an autosomal recessive rare genetic disease. An autosomal recessive disorder means that two copies of an abnormal gene must be present in order for the disease to develop. So the parents are perfectly healthy, but they are both carriers of a pathological mutation. In this case, the children will have the 25% of probability to inherit both the mutated genes and thus to have the disease. There are several forms of crab disease. The most common and severe is the early infantile form, with symptom onset within the first 6 months from birth. The gene which is mutated in crab disease is the GALC gene. This is responsible for the production of the GALC enzyme. GALC carries out its action in cell lysosomes that are specific cell compartments that contain many enzymes whose function is to digest used cell materials, such as lipids, carbohydrates, or proteins. GALC is just one of these enzymes. Errors in the gene sequence make the enzyme to be produced with defects, leading to drastically reduced functionality or not to be produced at all. GALC mutations cause the GALC enzyme loss of function. Thus, the GALC scissor cannot cut anymore its targets. This leads to the accumulation of an undigested sphingolipid called psychosin. Psychosin is really very toxic. Its progressive accumulation kills the myelin cells, leading to a widespread loss of white matter and to the following neurodegeneration. Now, to treat the crab disease, the most obvious option would be to reinstate GALC activity where it is needed. Of course, first of all, in the brain. The dream would be to systematically and periodically administer functional GALC to recover the required enzymatic activity in the nervous system. This might mean making periodic GALC intravenous injections or maybe just taking some Galsi pills every now and then. Unfortunately, this challenge is formidable because of the presence of the blood-brain barrier. The blood-brain barrier is like a wall that separates the blood flow stream from the brain. Passing through this wall is a highly regulated physiological process, and it is practically forbidden for big drugs or molecules like enzymes. So we need to find another strategy. Our approach is based on the use of nanoparticles. What is nanoparticle? Well, it's a sort of intelligent drug with dimension that can reach a billionth of a meter. Here you see a scale comparison. Just to have an idea, the dimensions of a nanoparticle are to those of a ball, as those of a ball are to those of the Earth. These nanoparticles are intelligent because they can be designed to have specific functionalities. In particular, they are typically composed by a core with the drug to be transported, and a shell with an external decoration that gives the nanoparticles the capability to interact with the host in a very specific manner. In our case, we want to make the nanoparticles to reach the brain, go through the blood-brain barrier, and release the cargo in brain cell lysosomes, where GALC is expected to perform its action. Going a little bit more in detail, the nanoparticles that we have proposed are based on PLGA. PLGA is a safe and biodegradable polymer that can be easily shaped into nanoparticles with the right size to go through the blood-brain barrier. Then we have found a way to load PLGA nanoparticles with functional GALC. Finally, we attached to the PLGA a so-called brain targeting peptide. This is a molecular unit that has a high affinity with specific proteins present in the blood-brain barrier. Thanks to this targeting peptide, when the nanoparticles are systematically administered, they are expected to bind to the blood-brain barrier and automatically be transported to the brain side by these proteins, and here released. Once in the brain, the nanoparticles will be taken by brain cells into lysosomes. Here they will degrade and release their cargo, that is functional GALC, to restore the overall brain GALC enzymatic activity. In the next few slides, I'm going to show you some results of this research. First of all, we have produced and tested our nanoparticles in vitro with the cells from the Twitcher mouse, which is the most used mouse model of Krabbe disease, and most importantly, with the cells from Krabbe disease human patients. First of all, we have demonstrated that the nanoparticles could be taken in by cells and that the GALC cargo is released to the cell lysosomes. That is just where the enzyme is expected to carry out its function. Then, after 24 and 96 hours of treatment, we measured the GALC activity with respect to control untreated cells that did not carry any pathological mutation in the GALC gene. The results of these experiments are summarized in these two graphs on the right. One is for mouse cells and the other for human cells. The blue lines indicate the level of GALC activity measured in cells from control mice and human donors with no GALC mutations. As you see, the activity measured in treated cells increased from almost zero up to over the values expected for cells from control mice or humans. In other words, the treatments totally rescued the GALC activity in Krabbe model cells. So, encouraged by this result, we tested our nanoparticles in vivo, meaning not just in cell, but in the entire animal model. Here, you see the study protocol. At the age of 20 days, the animals were systematically treated with the nanoparticles via intraperitoneal injections. After 4 hours, they were sacrificed, and the brains and other control organs were extracted for GALC activity. First of all, we directly looked for the nanoparticles in mouse tissues by microscopy. In these images, the red dots are the nanoparticles, whereas the blue shapes are cell nuclei. Nanoparticles were found in livers as expected because the liver is a well-known accumulation organ. This, for us, was a sort of positive control. Most importantly, as you see here, we could find the nanoparticles also in the mouse brains. Concerning GALC activity, these are the values measured for control mice not carrying any GALC mutation. The values obtained for healthy carrier animals were found were around half of the ones measured for control mice. As expected, zero activity was measured for diseased mice. For diseased mice treated with the GALC nanoparticles, we could obtain a very important increase of activity, up to the level of carrier mice. I would like to stress that these values of activity in carrier mice is sufficient for the disease not to manifest at all. The carrier mice are indeed perfectly healthy. Well, now, what are we doing now, and what do we still need? The answer is quite simple. We have to try to cure the mouse. To this end, we have found a treatment protocol made by repeated GALC nanoparticle administrations that can maintain high levels of activity in the nervous system of the mouse. At the moment, we are performing a complete enzyme replacement therapy treatment and measuring lifespan, behavioral function, and biomolecular markers of the treated mice. We hope to complete these experiments and the relative data analysis soon. Okay, now I leave the audience to Ambra Del Grosso, who is going to proceed with the second part of the seminar.
Hello. Sorry, 1 second. Sorry, I have to check 1 second. I need a few minutes.
Thank you all for your patience and stay with us. Sometimes the challenges of computer and sharing your screen.
I try to call Ambra to check with her what is happening. Just give us a few seconds more.
Sure, thank you.
Pronto? Marco? Sì.
Okay. So her problem is the same of the one I had. She had to log out and then log in again.
Very well. So we'll be waiting for her to come back on the panel, and hopefully she'll be able to share her screen.
Yes, yes. It was just a matter of the Mac OS. Now she is logging in again.
Yes, so we can see your screen
Okay,
now.
you can see. Thank you,
Yeah.
and sorry for the delay. So, okay, I put the presentation up.
Thank you.
Okay. Good afternoon to all. Thank you, Chair. Thank you, Marco, and thank you to ELA organizers for having invited me here to present to the families my research activity in the field of autophagy in Krabbe disease. So, first of all, what is autophagy? Autophagy is a word that comes from Greek, where auto means self and phagy means eating. In fact, it's a process by which a cell break down and destroy all the damaged or abnormal proteins and other substances in its cytoplasm, which is the fluid inside a cell. And then the breakdown products are recycled for very important cell functions. The process starts with the formation of an organelle called autophagosome, which encapsulates in the cell all the material which have to be degraded, the waste material. Then this organelle fuses with another organelle called the lysosome, which contains lysosomal enzymes. Finally, an organelle called autolysosome is formed, where the waste material is degraded by the lysosomal enzyme. But why we study autophagy in Krabbe disease? As you can see here in the upper part of the image, in yellow there is a normal cell with lysosomes inside which have the correct lysosomal enzymes. In the lower part of the image, instead, there is a cell with a lysosomal enzyme deficiency as a Krabbe disease cell, in which there is a deficiency of a lysosomal enzyme, which cause the accumulation of waste material inside the cell. But what is this waste material which accumulates aberrantly in Krabbe disease cell? As Marco told in the last talk, the most studied and known molecule which accumulates in Krabbe disease cell is psychosin. And as you can see in the image, psychosin accumulates into the membrane of the Krabbe disease cells, destroying many important cell activities. But we asked us, are there other molecules which accumulate in Krabbe cells? And we decided to investigate about molecules related to the autophagy pathway. So for this, we used the Twitcher mouse, which is a very useful mouse model for the studies about Krabbe disease, because these mice have a mutation in the same gene of the Krabbe patients, which is the GALC gene, and have also pathology which is very, very similar to the human one. In particular, we investigated about a protein called P62, and we very interestingly found that it is present in form of aggregates only in the brain of the Twitcher mice, both in the brain of the mice of 20 days of age and also in the brain of the mice with 30 days of age. Whereas these aggregates are completely absent in the brain of the healthy mice. In fact, if we quantify the number of aggregates, we can see that the number is very higher in the mice, in the Twitcher mice compared to the healthy mice. And we can also see that the number of aggregates increase with the age of the mice. In fact, we can see a higher number of aggregates in the mice with 30 days of age compared to the mice, to the number of aggregates that we can quantify in the mice with 20 days of age. But what is the role of this protein, P62? P62 is very important in the cell because it binds the material that have to be degraded, and it allows this material to enter into the autophagy flux to be degraded. This waste material, which is tagged with P62, can be misfolded proteins or damaged organelles or lipids or other. Moreover, there is another very important protein for the autophagy pathway, which is called LC3. LC3 allows the formation of the autophagosomes and is very important because we know that when LC3 expression is increased, there is an increased autophagy flux inside the cells. It means that there is an increased autophagy flux inside the cells. So we asked us at this point, how is LC3 expressed in our Twitcher mouse? And what we found is that LC3 is overexpressed in both the Twitcher brain and in the Twitcher sciatic nerve, demonstrating that the autophagy flux is increased in both the central and the peripheral nervous system of the Twitcher mouse. We also found the upregulation of P62 in the sciatic nerve other than in the brain. And so thus our hypothesis is that the high amount into the cell of the P62 aggregates cause an increased autophagy flux as a sort of response of the cell which try to degrade all the waste material tagged with the protein P62. And so we decided to test autophagy inducers in order to help the cell to restore a normal condition without aggregates. Thus, thanks to the ELA-funded project in which I worked, we could preclinically test into the Twitcher mice 2 autophagy inducers. Very interesting, also because they are both already FDA and EMA approved for human use. In particular, they are rapamycin, which is used as the immunosuppressor after transplantation, and lithium carbonate, which is used for bipolar disorder. What we did with our mice regarding lithium, we gave lithium with the drinking water to the twitcher mice starting from day 20 of age until the sacrifice. Very surprisingly, however, we found that lithium was not able to promote autophagy induction in our mouse model, and also P62 aggregates reduction, and was also not able to reduce the astrogliosis, so the inflammation. However, very interestingly, we found that it is able to modulate the expression of the protein beta-catenin, which is a very interesting protein because it's linked to cell survival. And so this finding deserves to be further investigated. Regarding rapamycin, instead, we obtained maybe more interesting results. We gave also rapamycin to the twitcher mice starting from day 21 of age until the sacrifice, always in the drinking water. And we found very interestingly that rapamycin is able to increase the motor abilities of the twitcher mice at selected time point of the treatment. In particular, we can see that the motor force of the twitcher mice is increased when the mice are treated with rapamycin at the time point 2 of the treatment, that is 10 days after the starting of the treatment. And also the motor coordination is increased in the twitcher mice treated with rapamycin at the time point 1, which is 5 days after the starting of the treatment. Additionally, we examined also the brain of the twitcher mice treated with rapamycin, and we found a decreased number of the P62 aggregates in the brain of these mice. In fact, as you can see here, the red point are the P62 aggregates, and they are absent in the brain of the healthy mice. They are a lot in the brain of the twitcher mice, and they are very decreased instead in the brain of the twitcher mice treated with rapamycin. With the green color, here we can also confirm that rapamycin induces autophagy in the twitcher mice. In fact, the green color marks the LCG expression, and we can see a very high intensity of the green color in the brain of the twitcher mice compared to the brain of the untreated mice. Then we also investigated about astrogliosis, which is a very important feature of the disease. In fact, it is linked to inflammation and is very high in crab tissues, as we can see from the green color in the image. Very interestingly, in the brain of the rapamycin-treated mice, astrogliosis is very low and is comparable to the brain of the healthy mice, demonstrating that rapamycin is able to greatly reduce astrogliosis in the twitcher brain. Finally, we found that rapamycin is also able to reduce the psychosin accumulation into the spinal cord of the twitcher mice. And we are currently investigating why it happens only in the spinal cord and how we can induce it also in other organs. So to summarize the rapamycin result, we can say that rapamycin has been able to induce autophagy in our twitcher mouse model, and has been also able to reduce the P62 tagged aggregates of waste material, which are accumulated into the nervous system of the twitcher mouse. Moreover, rapamycin is also able to reduce astrogliosis in the nervous system, both central and peripheral nervous system, and to little improve motor abilities at selected time point. And finally, it has also been able to reduce the psychosin accumulation only in the spinal cord. Now we still have to continue and finish these studies about rapamycin treatment in the twitcher mice, and then depending on the results, there may be the possibility to get in touch with clinicians to talk about any clinical trial of rapamycin. And this would be certainly be greatly facilitated by the fact that rapamycin is a drug that is already in use. However, the scientific community which work on Krabbe disease agree that a single drug or a single type of therapy can probably not be the right way to find an effective cure for Krabbe disease. So in our view, there is first of all the need of a long-term therapy which is able to restore for long-term the missing enzymatic activity, as can be bone marrow transplantation or gene therapy. However, this type of therapy is known that they need a certain time to engraft and create their therapeutic effect after the birth. And thus, a buffer therapy as the nano-earth therapy, which immediately restores the Galci enzymatic activity, can be crucial to prevent the early birth damages of the nervous system. And additionally, also complementary or supportive therapies can be needed to target all the cellular pathways which are secondarily altered into the Krabbe cells, as for example, autophagy. So this is the last slide. And first of all, I want to thank you very much, ELA, which gave us the possibility to carry on this research fellow project for the preclinical testing of lithium rapamycin in the Twitcher mouse, and which supported us also for the pilot nano-earth project and for the preclinical test, preclinical nano-earth testing. And then I would like to thank my research group at the NEST laboratory of Pisa, that is called the Neuroscience Group. A special thank to Marco Cecchini, my PI, and to Ilaria Tonazzini, and also to Maria Cristina, Sara, Miriam, Laura, and Luca for animal studies and nanoparticle synthesis. And finally, thank you all for your attention.
Thank you very much, Dr. Del Bosco. Thank you also, Dr. Cecchini, for this presentation. I will just introduce very briefly our next speaker because we are a little bit on schedule. So Dr. Caroline Sevin is with us, and she will talk about the management and the treatment options for Krabbe disease and metachromatic leukodystrophies. Dr. Sevin, the floor is yours. Thank you.
Hello, and thank you so much. I'm going to try and share my screen. Hopefully it'll be easier than before. We can see your screen at this point. Can you see my screen? Absolutely. Well, good afternoon. Good evening.
Donc, je vais faire cette présentation en dernière position.
So I'm last. First of all, I would like to, after all the other presenters, very high quality presentations, by the way, I would like to first thank all those of you who accepted to stay with us. I work as a neuropediatrician at the Kremlin-Bicêtre Hospital in Paris. I'm also at a reference center for leukodystrophies. Today, I would like to talk about two leukodystrophies that are much more frequent in children than in adults. We're going to be talking mainly about pediatric considerations, and even though sometimes some of those pathologies will appear in older subjects. So some of the information I will be delivering applies to children, but also to adult subjects. And some treatments that are not available at this point to adults, I hope will be available for them in the near future. So MLD and KD, Krabbe's disease, have many things in common. First of all, they're lysosomal storage diseases. We have already heard this in previous presentations. Both pathologies are mainly limited to the central and peripheral nervous systems, so that's brain, spinal cord, and peripheral nerve. Even if sometimes there are other parts are also affected in around the brain. we see rapid degradations in early onset forms. And so we are talking about leukodystrophy with demyelination, but also neuronal degeneration and neuroinflammation. We'll come back to that a bit later. So for early onsets, when symptoms are present for both pathologies, no treatments exist. They are the most frequent ones. For later onsets, especially, or if the hematopoietic stem cell transplant can help, ex vivo gene therapy exists. It was developed by Orchard Therapeutics for children who are screened and detected before the symptoms. And for them, things can really be changed. And there are some additional trials that are underway for both pathologies. So a quick update on MLD first. MLD is one of the most frequent pediatric forms of leukodystrophy. It is caused by the mutation of sulfatase A, which degrades, normally degrades lipids that are very important in myelin. They're called sulfatides, and by accumulating them, they will eventually cause lesions and the death of oligodendrocytes, as well as other cells such as neurons, for example. So the sulfatide accumulation will cause symptoms, leukodystrophy and clinical symptoms, which is, there are some clinical signs that will occur within a very specific age bracket, and they should be warning signs for physicians, and at MRI as well. You can see the peripheral nerve is affected, for example. You can see it at the speed of nervous conduction, for example. To confirm diagnostic, we need to check enzymatic activity, sulfates in urines, and additional genetic testing. As I said, we're seeing a lot of early forms, early onsets. The pathology can appear at age 1 and all the way through the adult age. The classification is true generally for lysosomal diseases. Generally, we see, the late infant one is the most common one, 60% in France. It's not necessarily the case in other countries, as a matter of fact. And then we have the juvenile early, between 4 and 6, and later forms, late juvenile and adult. The ones that are in red are the most common ones. Once again, we're seeing rapid progress with generally the same symptoms: walking, disability, hypotonia, ataxia, severe neurological impacts, cognitive degradation. In later onsets, progress is slower. The first signs that should alert a healthcare professional should be cognitive signs that will generally precede motor signs. For Crab disease, again, it is also a lysosomal storage disease. It is a more rare disease, as we've seen before. This is it is a deficiency in GALC, G-A-L-C, which creates the accumulation of galactocerebroside and psychozyme. Here again, the early infantile form is the most common one. It starts between the age of 6 to 12 months. There is a little bit of a disagreement on that classification, but early onset is generally before age of 6 months. The first symptoms appear through discomfort, stiffness, very characteristic, and pediatricians will generally seek and struggle before they will identify the pathology, and generally death will follow within the 2 years. Then we have the late infantile form, 12 to 36 months, and other late forms, juvenile and adult. For those, we are seeing motor delays, we're seeing psychomotor regression, and additional symptoms that will appear for late forms. The juvenile and the adult one, progress is slower. We're seeing spastic paraparesis, cerebellar disorders, visual disorders, with a lot of variability from one family to another. What the two pathologies do have in common is that they're oftentimes diagnosed too late. With Crab disease or MLD, the evolution, as you can see, we have what we call a free interval during which the child will develop normally before stagnating. If you're noticing more progress, motor progress, and eventually regression, this is, obviously why the earlier, the better. Ideally, the treatment before symptoms would be better. During the stagnation period, it's still okay. But if you've reached the regression phase, the disease will progress very, very rapidly and making treatments much more complicated. And generally, a single treatment won't work. You need to do combined treatments. And for MLD, in first symptoms in later forms are between 15 and 18 months, and generally takes a year to be diagnosed. So that is very, very long. And generally, when you see patients at the time of diagnostic, the disease has already made significant progress. A few words on the physiopathical mechanisms. The physiopathological mechanisms are very well known. Sulfatides accumulation on one side. Same thing for Crab disease. This accumulation causes cell toxicity, which will eventually cause major changes in secondary mechanisms. We've heard this in the previous presentation with different types of anomalies that can be seen. Oxidative stress, for example, which eventually causes death cell. And there is another very important mechanism in these, in both pathologies. It's neuroinflammation. It's not only true for MLD and Crab, it's also true for a lot of other lysosomal diseases, storage diseases. With both pathologies, MLD and Crab, we have seen and found that the neuroinflammation is not necessarily secondary or caused by the accumulation. It can actually sometimes precede demyelination. So it plays an important role in the physiopathology. And when it comes to treatments, this is something that should be taken into consideration. Moving on to treatments. We're going to go through some of the treatments that exist. There are different treatment pathways. And what was again said very clearly in the previous presentation is that one single treatment will, in all likelihood, not be enough. And in most cases, we will need to combine treatments. In clinical trials, you can't combine. But in the future, we will need to combine different treatments. Some treatments have shown partial efficacy. It doesn't mean they don't work. If you combine them, potentially with combining them together, you can get better results. So before we talk about innovative treatments or specific treatments, I would like to say what Mark Engelin said in his presentation as well, is that the absence of curative treatment doesn't mean absence of care. improving
la qualité
medical
de vie
care,
des enfants et
improving
des
quality
patients
of
ne doit
life
pas
of
arriver
patients
ne doit pas être la volée
is
de traitement.
not
Il faut
something
traiter
that should
dès
be
les
done
premiers
when you start
symptômes, donner
treating.
des
You have to
traitements
treat as
d'antidouleurs
soon
rapidement.
as the first
Donc,
symptoms are
ces
observed.
signaux cliniques qui
Stiffness,
sont à la fois
all the clinical
invalidants,
signs
parce qu'ils diminuent
are
la qualité
invalidating,
de
first of
l'enfant,
all,
parce que
because
c'est
they have a
très
negative impact on
difficile de
quality of
voir
life of
ces
children.
enfants
It
qui,
is
en
very
particulier dans la
difficult to
maladie
see
de
those
Crohn, mais
children,
aussi dans la
especially with
dermatite
Krab's
atopique,
disease,
sont
but
très
also with
loin
MLD. They're very painful
d'arriver à
symptoms,
donner des
and
traitements, et plus tôt on les
finding
donne,
treatments is,
il faut
is
pas
difficult,
hésiter à donner des
but
traitements de la
it
douleur.
is obviously really
Plus
important to
on
treat
pourra
early,
préserver
including
à la
to
fois le
treat the
confort, mais
pain,
aussi
to preserve their
le
quality of
fonctionnel
life,
de
and
ces
also
enfants.
to
La deuxième chose, c'est qu'il faut s'adapter aux
maintain
traitements.
functions.
Il faut
Then
s'adapter aux
it is
symptômes.
important to
C'est-à-dire que
adapt to
dès qu'un
symptoms
symptôme
and
apparaît, il faut
adapt the
traiter.
treatment
Dès qu'un
to
autre symptôme
symptoms. If a
apparaît, il faut
symptom
traiter. Il
appears,
faut
it
pas
should be
hésiter
treated.
à
If
donner un
another
nouveau
symptom
traitement.
appears,
Beaucoup de
treat
parents
it as well. Don't hesitate. Many
préfèrent
parents
attendre un petit peu,
would
en
rather
particulier
spare the
pour la
child,
raideur, pas pour la douleur, parce que pour la douleur,
not so
on
much for the
on
pain,
peut pas
but for
voir
stiffness.
un enfant souffrir, mais
Having
vraiment adapté, c'est vrai aussi pour tous les
children
matériaux,
who
toutes les
suffer is
prothèses,
not acceptable. But
les choses, les
in
attelles,
addition to that, I
tout ce qu'on
really
va
think
mettre
that
en
all
place, il
other
faut
treatments are
vraiment aller
necessary
très vite.
to help children remain,
c'est vrai
keep their
que
mobility, walk, and so
on
on.
a tous ces symptômes, la douleur, la
So a
raideur,
lot of symptoms:
l'épilepsie, les
stiffness,
problèmes
pain,
qui surviennent un
epilepsy,
peu plus tard dans la maladie, les
breathing,
difficultés respiratoires, les troubles de
swallowing
déglutition.
disorders.
Il faut favoriser la motricité.
It is
Ce sont souvent
important
des
to
questions aussi qui sont posées.
encourage
Est-ce
and
que je
favor
dois
motricity.
le laisser complètement tranquille ou stimuler? Il
So
faut stimuler, bien sûr.
if the question is, should
L'enfant
we
va
just
donner
let
ses
the child be
limites.
or
S'il est
stimulate him or
douloureux,
her?
bien sûr, il
He
faut pas
should be
insister, mais
stimulated,
il faut
unless,
vraiment
of course, it causes
arriver.
excessive
On a vraiment
pain. But it is
des
important to
parents
promote
et
motor
des
skills.
rééducateurs qui ont
And
plein
we
d'idées
have
pour
parents who are
avancer
very
dans
inventive with lots of
point de
ideas.
vue-là.
We have
Et puis, il faudra une prise en charge
healthcare professionals who
multidisciplinaire.
have
Je
lots of
vais pas
thoughts,
redétailler parce que
great
c'est
ideas as well. It is
vraiment
important to provide multidisciplinary care,
décrit
as
par
described
Fred Hélène, mais clairement, c'est
earlier
pas
again by
uniquement
Mark
un
England.
neuropédiatre ou un pédiatre. Il y a
Children should not
vraiment
just
une
see a
équipe
pediatrician or a
autour de
neuropediatrician. They should
l'enfant
see,
pour,
you
une
know,
fois de plus,
a
améliorer
true
l'enfant, mais aussi
multidisciplinary team
donner à l'enfant la possibilité
to
de faire
provide
le
the
maximum
child
de ce qu'il
with,
peut faire
you
en
know, to
termes
do
de
as much
motricité, en
as
termes de
possible
langage, en termes
in
d'interaction.
terms of
Ça
language,
demande donc un suivi
psychomotricity,
multidisciplinaire, un
and so
soutien
on. So this
psychosocial. Et
multidisciplinary and
ces enfants,
psychosocial
le plus
support
souvent,
is
doivent être suivis dans
absolutely
des centres de
essential.
référence,
Most
au moins en
children have to be
alternance,
seen
mais
in reference centers
des hôpitaux et des
at least once
neuropédiatres
every
proches de
2
chez eux,
visits,
mais c'est important aussi la
if possible,
communication
in
entre
not too far away
les
from where they
centres de
live. But
référence
having
et
good
les équipes
communication
qui
between
suivent l'enfant
the different
régulièrement.
healthcare professionals and experts is essential.
Donc, quel est le traitement idéal? Donc, vous avez
So,
vu tout à
what
l'heure,
is
je vous
the
ai dit
ideal
cette
treatment? Again,
répartition
earlier
entre
on, I
évolution de la maladie avec la phase
told you about the onset,
présymptomatique, la phase
the
vague
pre-symptomatic
symptomatique de
phase,
stagnation,
and the early
la
stagnation
régression
phase, the
rapide, et puis
symptomatic
des
regression
symptômes tardifs qui peuvent apparaître.
phase,
Et
and
clairement, prévenir est plus facile
late
que de
phases.
réparer.
It
Aujourd'hui,
is
on ne sait pas
obviously
réparer.
important.
On entendait
We
déjà,
don't know
c'est
how to
pas
repair.
facile.
So if you, the more
Réparer, ça va
advanced
être
you are, the
très
more difficult it
difficile.
becomes to
C'est pas
repair.
remplacer des cellules.
We
Peut-être un
cannot
jour, on
replace
saura, mais
cells.
pour le moment,
Maybe
c'est
we
quelque chose
will. Right
de
now,
très
we
compliqué.
can't. It's very
Les
complicated.
traitements dont on va parler,
The treatments
tiennent compte d'une
we're going to
propriété
discuss
de ces enzymes lysosomales, qu'elles
are
sont
based on
capables
these
en fait
lysosomal
d'être
enzymes, and
sécrétées par une cellule
they can be
et
secreted by a
recaptées
cell,
par la
and
cellule
they
d'à
can be
côté.
recaptured
C'est-à-dire
by
que
the
quand on
cell
va par
right
exemple
next
faire
to
de
it.
l'enzymothérapie,
So
quand on va donner l'enzyme dans le
when you do
sang,
enzyme therapie, enzyme
elle va
through the
arriver dans le
bloodstream,
cerveau,
when it hits the
elle va
brain,
devoir être captée par
it's
les
going to
cellules. C'est
be
indispensable,
captured
sinon
by the
ça
cells,
sert à
which
rien.
is
De la
indispensable.
même
Otherwise,
manière,
there would
quand on
be
va
no
faire une
point.
thérapie génique, on va
Likewise, when you do
corriger
gene therapy,
génétiquement
we're
une
going to
cellule dans le
genetically
cerveau, on ne
correct
va
a
pas
cell
corriger
in the
toutes
brain.
les cellules
We're not going to
génétiquement, on
correct
va corriger une partie
all the
des cellules,
cells. We're going
et ces
to
cellules
correct
vont
some
pouvoir
of them,
comprendre
and
la sécréter
those
et la
cells will
diffuser aux
be able
aux cellules
to
voisines.
then communicate and
Donc, qu'est-ce
pass
qu'on peut
on
faire
this information
si on
to the
regarde
neighboring cells.
sur ce schéma? En fait,
So
on peut remplacer
let's take a look at this slide. You could replace genes by administering the genes, either IV, IC, or it will then enters the cells. This is in vivo gene therapy, or genes could be transplanted. These are hematopoietic stem cell transplants, the donor cells will then migrate into the brain, and they will make normal enzymes, and they will deliver those cells, and they will create the enzyme, which will then be recaptured by the neighboring cells. Once again, one could also replace the enzyme itself. So it's called enzyme replacement therapy. In MLD, there is a clinical trial ongoing. We could also manage secondary mechanisms, like oxidative stress, for example. These molecules also have an important role to play, especially when combined with replacement or like enzyme replacements. Moving on now to more specific treatments. First of all, we have transplants, allografts either conventional ones or genetically corrected ones. So the donor cells will migrate to the brain, as I said before. They're going to make the enzyme that is deficient, give it to the neighboring cells. It has an impact on neuroinflammation as well, because the wrong cells will be replaced by non-deficient microglial enzymes. And this is a very important mechanism. Inflammation is a major physiopathological mechanism. The treatment is complex. It requires ablation. You need to empty the marrow in order to then allow the cells to enter the bone marrow. It is a very long treatment. It takes 9 to 12 months to really get the full benefits, because replacing, especially in the cells, especially in the brain, will take time. So then we have the hematopoietic stem cell transplant with genetically corrected. We take the cells of the child and we take them to a lab, and you can actually correct them, genetically correct them before re-administering. So it's an allograft, it's an auto transplant, which requires myelinoablation. You need to empty the marrow and before re-injecting it in the marrow. Otherwise, the effects or benefits seem to be higher and faster as well. The reconstitution, which is the time it takes to all cells to recover full function with the blood cells. No need to find a donor, which is obviously important. In addition to that, the cells that have been corrected, instead of simply expressing the enzyme, they will overexpress it, because you can put several copies of the vector to obtain an overexpression of the enzyme, which potentially also have a positive impact in terms of efficacy. Okay, let's come back to the allogenic transplant with a donor. Currently, this is a treatment which is available for MLD and for Krabs disease, but in specific contexts only. In MLD, it can be discussed for late forms, juvenile and adult. The transplant will modify the journey, will change the history of the disease. But it should be done early, as soon as clinical symptoms, clinical signs appear, possibly even before, and it doesn't fix all the problems. It won't keep the disease from progressing. some, the cerebellus, for example, or cerebellum, sorry, will not be treated well. It is not a perfect treatment by no means. It should be done. Transplants are necessary, but there is probably a need for complementary treatments to really optimize it. For other forms, if the children are treated pre-symptomatic, it changes the natural history, but it's not sufficient. It's for early symptomatic forms, once the symptoms have appeared, the treatment is not recommended because it has no effect. Probably because of the very fast evolution of the disease, and again, the transplant will take several months before it becomes active. For Krabs disease, it's a bit the same. For late forms, the treatment can be offered either pre-symptomatic, that's before symptoms appear, or the very early stages. For the early infantile form, the treatment is only recommended for pre-symptomatic children. So that's the first month of life. Earlier on, I mentioned 3 months it takes, so it should be done very, very early. And even with children who receive a treatment very early, if the transplant works well in the long run, we still see motor symptoms, especially because the peripheral nerve is not always treated very well following the bone marrow transplant. So transplantation does make a difference. There have been different tests and trials of hematopoietic stem cells, especially in MLD. A treatment is now being, is available in a number of European countries in an early access. It is being discussed to determine the price before it can be commercialized, but children can be treated. So this trial was conducted in children who have an infantile, late infantile, pre-symptomatic form of the disease, not in symptomatic children. It doesn't work. So it's pre-symptomatic children only, and in early juvenile forms. So that's the beginning of symptoms, age 2.5 up to 6 years, or pre-symptomatic children, or children who had very mild symptoms. 29 patients in total received the treatment, 16 late infantile form before symptoms, 13 juvenile, early juvenile, either before symptoms or at the very beginning of symptoms. So let's now take a quick look at the results. First of all, the tolerability is good. No specific problems were reported. Obviously, the chemotherapy is complex. The treatment overall is complex, but tolerability all in all is good, with very few or no severe side effects. Then it was found that the treatment allows to restore enzyme activity in the blood, with the blood cells, and especially in the cephalorachidian liquid. There's a lot of enzyme activity there as well, which is very present and again observed at more normal levels. There is also a significant effect on the preservation of motor functions, as you can see on these two charts at the bottom, at year 2, year 3, post-treatment in children with late infantile forms, pre-symptomatic, or children who have early juvenile forms. We can see that the impact is very substantial. It preserves motor function. We use the GMFM scale. Maximum is 100 at age 4 or 5, and as you can see, non-treated children have pretty much lost all motor function. Those who've been treated have preserved much better motor function, and this is a result which is very, very positive. after two years or 3 years. The treatment also allows to maintain cognitive function in both forms. In gray, you can see the normal cognitive evolution. You can see that the children are very close. The ones that are at the bottom here in light blue or light green are those who have not received the treatment, and you can see the cognitive degradation is very severe. On the right side, it also prevents leukodystrophy in those who've been treated pre-symptomatically. That's before lesions. It was able to stop the progression in those who already showed some symptoms. So the bottom line is this is an essential treatment in metachromatic leukodystrophy. Again, only if several criteria are met: pre-symptomatic, or early juvenile, pre or early symptomatic. There is approval, European approval was given, and the treatment is now accessible throughout Europe, especially for early in early forms. In France, we've been able to treat since October 2021. We've treated 3 patients so far. And for late juvenile forms, right now it's not available, but there is a trial in progress for pre-symptomatic childs or early symptomatic children. Sorry. I talked about enzyme therapy a bit earlier. There is an interesting one which is ongoing, where the enzyme is administered in the liquid, the encephalorachidine liquid through a catheter. It is a complex treatment, but tolerability has been good. Once the optimal dose is found, there are no side effects, and the higher the dose, the better the results. The results of this first study have also shown the normalization of the sulfatides in the CSF, and there is another phase 2-3 trial which is underway with an injection, not every other week, but every week, 150 mg. The results should be available by the end of 2023 of this year. What I can say at this point, if that this treatment has an impact, but it is not enough to stop the disease. So it is an interesting treatment. I hope it will be approved and eventually commercialized, but it will probably have to be combined with other treatments. Moving on to Krabbe disease. Earlier on, we talked about the hematopoietic stem cell transplant for early infantile forms. Even if they've been treated pre-symptomatically, there is an impact, but it does not prevent long-term development of motor deficiency. And David Benger's team was able to demonstrate in mice, which is a great model for the disease, that when you transplant and you add gene therapy, IV gene therapy, you can obtain superior results to transplant only or IV injection only. By combining the two treatments, you will significantly improve the outcomes. You can see the survival of mice in blue. You have the combination in purple, no treatment, and green and orange is those who received one or the other of the two treatments. So you can see that alone, you get some improvements. If you combine the two in terms of survival, the benefits are quite impressive. Same thing for motor disorders. It really corrects the clinical phenotypes of in mice, and it corrects the neuroinflammation. Likewise, it also allows to, the prevention or protection of the peripheral nerve. So these are very positive and promising results that eventually led to another clinical trial, which is currently taking place in the US with Forge Biologics. So there are 2 trials that are happening at the same time. one with a transplant followed by gene therapy in very symptomatic children. So they have to be symptomatic, they've been diagnosed, they will receive the transplant as described earlier, very early, and within 20, 30, 60 days, they will receive the injection. And so that's the first trial. The second trial, which has just started recently, is for children who've already been transplanted a while ago to receive one injection for the gene therapy, an additional one, even if they've received the transplant more than 90 days ago. So this also shows that children who've received a transplantation will potentially be able to benefit from this additional treatment, in particular to protect the peripheral nerve. So this is it. A pretty fast overview. I have not even talked about other treatments that are being developed, but I think some of the bottom line messages is that new treatments do exist for MLD and for Krabs disease. These treatments have to be administered ASAP, possibly before symptoms in some patients. When you have a child who's diagnosed and who is already showing symptoms, treatments are not available, obviously. But I think this also shows that screening is very, very important in families. The younger siblings, brothers, sisters should be screened very. Neonatal screening is absolutely essential. It's going to happen eventually in France and in other European countries. It's going to happen eventually. We as healthcare professionals, researchers, we have to keep in mind that kids should be screened with the very first symptoms. We have a screening problem. Once the diagnostic, once you start screening, you can get the results very quickly, 1-2 weeks. It must be done quickly. And last but not least, we also need to better understand the very early symptoms. And with MLD especially, there is an international register which exists to actually understand the disease, identify the disease before the very first symptoms. When there is no neonatal screening, that is what should be done instead. I'm going to leave it here. Thank you so much. I'd be happy to pick up your questions. Thank you.
Thank you very much, Dr. Sauvin. Indeed, if you'd like to ask a question in the audience, you can raise your hand on your screen, at the bottom of your screen. We are beyond schedule, but if you, we can take the time to answer a few questions. So don't hesitate. Otherwise, you can also ask them in written, and we'll answer them after the meeting. So I do not see any raised hand yet. Very well. So I think maybe everyone is tired from these very intense, very rich presentations. Then I guess we conclude. I would like to thank all our speakers for today, and hope we can answer.
Excuse me, I think there were questions that I didn't answer in the
writing
in the Q&A.
If you'd like to answer them, either we can answer them orally now if you have a minute, otherwise we can address the answer to the person in writing after the meeting.
Well, as you want, but I didn't answer directly to Patrick Winters because I found they were very important questions. So,
okay.
Well, if we have time, I can quickly answer them. Otherwise, I will do in the.
We can. If you can read the question first and then answer, that would be fine.
Yes, because one important question, it was what has been done, if anything, to confirm or deny the hypothesis that RG, so a retrospective inhibitor, could be effective for ADA1 and EPH1 related AGS, etc. So I wanted to say that what we have understand so far in the underlying pathogenesis of ADA1 and its ventral mutation, there is an indirect, it's not directly to the nucleic acid, so that's why there is no hypothesis that this drug can be effective for these two mutations, because they are not related to the detection of retro element, but to the editing that is not done by ADA1. So there is no hypothesis that the drug could be effective for these genotypes. And then the other important question was about the JAK inhibitor. So how can we reconcile the positive development trajectory of AGS patients treated with JAK inhibitors with the belief that it doesn't have a therapeutic effect on the brain? So this is a very important question, and it's not easy to answer, but the few important point is that as we all know, AGS is a severe disease, mainly affecting very young children. So when the drug was started, there was already an already established brain damage, both in the trial done in the US or what we have done here in France at Necker Hospital. So this is the first point. Then we know that in this disease there is progression, and that some patient can gain some milestone even without any treatment. So this is complicated oh, sorry, I'm a bit tired. I don't find the word in English, but the assessment of the drug effect. And the other thing is that there is a phenotypic variability, and some patients are less severely affected, and so they are even more able to gain some milestone. And our experience here in France is that there is no big gain with the JAK inhibitors on the brain involvement. There is clearly an effect on the skin, on the systemic involvement of GSDS, but unfortunately on the brain, we are not quite sure that there is an effect, although a patient did gain milestone under the treatment, but it could have been just the disease natural course.
Thank you very much for these precisions and these answers. I will read another question from the audience also regarding MLD. There is a question. Wait, Caroline.
Yes, oui, I speak in, oui, je parle en français surtout.
Yes, I will be speaking in French. The question is, what changed to say that neonatal screening of MLD can arrive in Europe? And so far, we can't really say if this will be able to happen in a year or 2 years, but what we can say is that now there is a treatment that can be carried out for only pre-symptomatic patients, and this treatment has to be administered very early on, so in the first few months of life. And it is a very important criterion when we ask for a neonatal screening, especially for a disease that is quite frequent amongst rare diseases. Secondly, there is also mobilization in Europe to move forward, and maybe it wasn't the case a couple of years back, but we are trying to have an European initiative and not a European initiative country per country. So it's hard to change our institutions, but hopefully we can have a European initiative, and pilot studies will be carried out in different European countries and not only in France, and hopefully with these different pilot studies, we will be able to move forward. Thirdly, I believe that, I'm only speaking from a French point of view. I know that it might be easier in other European countries, but in France, it's very hard to have a neonatal screening done to screen for diseases, where in a country in which very few screenings are done. But hopefully with new treatments coming onto the market when it comes to leukodystrophy or other types of rare diseases, with the arrival of these new very efficient treatment, hopefully we will be able to move forward and move quicker forward. I cannot give you a time frame, but there are talks underway, pilot studies underway, and other European countries are probably already
Thank you
further
very
ahead.
much. There is no more question, and I think we have reached the end of our time. So I would like to thank again all our presentators and our translators as well, team behind the screen, for this fantastic meeting. And I wish you all a very good night. Thank you. Thank you. Bye.
Thank you very much. Bye, everyone.